So welcome everybody to this, the second day of the Physics of Brains workshop that has been organised as part of the programme of activities in the UKRI and Rose Trees Trust funded Physics of Life and Medicine network. For those of you who weren't at the first day of the meeting yesterday, I can promise you that if the second day is as good as the first, we're in for a treat. But before moving on to the programme proper for today, it's perhaps worth noting that we have a follow-on workshop to the Physics of the Brains meeting, which is on February the 10th. And it's a workshop looking at neurodegenerative diseases of the brain. And again, in common with all these workshops, it's deeply interdisciplinary. We're absolutely focusing on questions to which we do not know the answers, and for which a truly interdisciplinary perspective might lead us to some really novel approaches and solutions. And as you can see, we've managed to engage with some of the more clinically facing research funders here, and I'm delighted to say they're really enthusiastic about this whole programme. The programme for today is hopefully well known to you. We've got two sessions. We'll have a 5-minute comfort break between the two sessions. And I would strongly encourage you to use the Q&A function on Zoom, which you will see. The Q&A tab is at the bottom of your Zoom window. And what we'll be doing as chairs and panelists is keeping a careful eye on what's coming through there. And we will ensure that you get your A to your Q, either through the panel discussion or more directly from one of the speakers. The final point I want to draw your attention to before we go to the first speaker is the Wonder session. Now, this is a terrific platform, and I speak as something of an IT cynic, one might even say, but the intention here is to bring people together in a virtual meeting room configured as breakout groups, each of which will relate to a set of questions that emerge both from today's work and the discussions we had yesterday. So more of that anon. Without further ado, we'll move on now to our first speaker for the session, which is Fred Wolf. Fred is from the Max Planck Institute at Göttingen. And we're looking forward to Fred's presentation on neural circuit dynamics. Fred.
Thanks, Steve, for the nice invitation and introduction and the invitation to present here. What I want to do today, in the spirit of this workshop, is to focus on things that, on the one hand, are promising novel, hard to answer questions, and on the other hand, for me, illustrate how working on the brain can actually give us fun physics in reverse. Specifically, what I'm going to talk about is the link between biophysics and the dynamics of the large networks that we all want to understand ultimately. So thousands, millions, many millions of neurons working together. What they do in cognition and in neural computation certainly depends on the finest detail of their molecular makeup. When we look at them as dynamical or statistical physics problems, they are huge dynamical systems. And so one fundamental question is how issues of stability and control, different phases of dynamics, come into play in understanding brain circuits. So one thing that's really fundamental here that I want to start with is to point out that nerve cells actually speak two different languages. What you're seeing here is a recording from a nerve cell in the cerebral cortex of an animal. The continuous curve is the membrane potential, the main physical quantity with which this nerve cell calculates. And on top of that are what you all know are nerve action potentials. Those are the signals that actually are communicated to other cells. They are tiny electrical pulses, as you see on when you look at the scale bar. And importantly, they happen fairly rarely. So the scale bar on the time axis is a second, and you see that barely there's one spike during a second. That is something that's really fundamental when you want to think about what is the information carrying unit in brain networks. Here we're looking at a single reconstructed neuron from the cerebral cortex. Indicated is its AD converter, where the place in the cell where continuous intracellular voltage signals are converted into spikes that travel to the rest of the brain and eventually control behavior. And because we know from many studies over the past 10 to 20 years that these spikes are so sparse, it is basically a set thing that the individual neuron cannot really carry information on the behaviorally relevant timescale. The relevant dynamical variable is the population activity of large groups of such neurons. So this conversion from continuous signals to discrete pulses that are sent around in these networks, you may think that is biophysics 101. There's a British Nobel Prize for it. Hodgkin and Huxley got that one. Isn't it a completely done deal? And so I want to briefly mention, and I will mention again, that this is for the nerve cells that we are using in our brains, not at all settled biophysics. And the reason, if I want to give it in a very pictorial fashion, is this one. What you see here is the biological system from which we learned the dynamics of action potential generation, the squid giant axon. And it's really it has been, and it still is a giant axon, so that Hodgkin and Huxley at Cambridge could put a wire down the middle of this nerve cable. It's so big that an entire nerve cell in your brain will easily fit into that. So we are trying to extrapolate our understanding of action potential generation like by 3 orders of magnitude. And that is the reason why the biophysics of nerve impulse generation is still a very active field. And I show you in a brief mention how that is actually an active zone where neural computation and biophysics, there's a back and forth that actually hopefully produces new biophysics. So from a statistical physics perspective, the problem that we're dealing with in order to understand and describe the action of a population of cells is to write down a stochastic model for population activity where there of every individual cell, there's some synaptic background fluctuations, and there's external input that this population is processing. There are basically two approaches to get traction on this mathematically. One is, since this is a stochastic differential equation or a system of those, we can use transport theory, Fokker-Planck theory, to calculate probability currents, and those probability currents in this Fokker-Planck equation are actually the firing rate of the population. So understanding its dynamics is what gives us information about the bandwidth or the filtering properties of a neural population as it processes information. The other one is using Rice equation. They have their advantages and disadvantages both. So the Rice equation based theory where you just write down as an expectation value over a population of cells the population firing rate as is here in the lowermost equation. You can easily handle arbitrary correlation times, while Fokker-Planck approaches kind of work best when you feel comfortable with the diffusion equation with a very short time background noise correlation time approximation. So what I'm going to show you in a few applications is that already in first order perturbation theory, when we assume that the information that's impinging on the population is just a tiny perturbation of the background noise that impinges on every neuron in this circuit, is fairly interesting. So I'm going to show you one example of such a study. We did it a few years ago, following up on a paper that highlighted that the first sub-millisecond in the life of such a nerve impulse can be a bottleneck for neural information encoding when it comes to information rates. Was this one. So here you see in the uppermost panel the simplest approximation that you can make for a spiking neural dynamics. So there's in this v.v plot, so it's a phase plane if you want. The origin is a stable fixed point, and there's another not unstable fixed point, and by their slopes we have control in this model over the stability timescale and the instability timescale of this neuron. What we found, if you know Fokker-Planck equations a little bit, perhaps not surprising, is that dealing with this instability has a huge impact on the bandwidth of neuronal encoding. So what's graphed here is the linear, the susceptibility of this population. How much can you vary its population firing rates when you put in an external signal of a given frequency? And this susceptibility can become completely flat and broad bandwidth the more unstable we make this fixed point. So on the right hand side, you see the time domain representation of these results. So with a high instability of the single neuron dynamics, you get an instantaneous response, which is equivalent basically to a very fast, to a very high bandwidth susceptibility. Here are a few measurements where we took living neurons in collaboration with Maxime Wolbushev's lab and turned them into a system of stochastic differential equations. So we emulated a live neural population in this fluctuation-dominated firing regime. And to our surprise, we found that basically there is no bandwidth limitation. This population could switch between states on a sub-millisecond time scale. So that result has triggered interesting questions in the direction of what is really going on with the action potential generators that we are using, what is setting their time constants. And while experimental research so far has confirmed and confirmed, again, this very broad bandwidth and the ultra-fast response of neural populations, what the underlying biophysics is still a matter of ongoing research. So one hypothesis, the one with which we started this business, was to show that once you make ion channels in the nerve membrane in this spike generating region of the neuron cooperative directly, so that the molecules can switch each other open, that is a way to achieve this very high bandwidth. There are two alternative theories that are currently being followed up, and if I had more time, it could be an entire talk to go into this back and forth between the bandwidth of information coding when you look at the population as an information processing device and the very fine molecular architecture of this subcompartment of the neurons. What I want to do with the remaining 5 minutes is to switch gears and switch perspective and take you through a few results that show you how the very same qualities in the single neuron dynamics, the very same features of the single neuron dynamics that make a population in this sense high bandwidth deeply impact on the dynamics and the phase space organization of recurrent networks, the interacting systems that we ultimately have to understand. So I'll do that with another simplistic model like the one I showed you. But here we didn't glue together two linear approximations, we glued together two square functions to build voltage dynamics, a single neuron model voltage dynamics. The advantage that is it has finite time blow up when you go here beyond the spiking threshold, which makes it unnecessary to introduce any reset or threshold reaching conditions that can induce artifacts, and we want to avoid them. But long story short, there's a parameter in this model that while it doesn't move the resting state of the single neuron and the unstable fixed point of the single neuron, it can make this unstable fixed point very, very unstable and the spike onset very sharp. That will also be relevant in this noise-driven naturalistic regime, and it will leave the same impact on the activity of an entire ensemble. So with the more sharp spikes, with the same number of action potentials, a population of cells can follow tiny and fast modulations of an input much more faithfully than with a sluggish onset of the action potential. When we look at the information rate in an individual population, well, it increases with this, making the individual spike sharper and more precisely localized in time, but it's a factor of two question. It's not a qualitative effect. The big surprise to us happened when we looked at entire recurrent circuits, and recurrent circuits in this fluctuation-driven regime generically are in a chaotic state. So it's interesting quantify them in the first pass analysis by Lyapunov exponents and by entropy rates. What you see here is the largest Lyapunov exponent of a recurrent circuit for 3 different levels of activity as a function of how sharp the onset, what is the time scale of this the shortest biophysical time scale in the system. And you see that, so for low values of this parameter, maybe that is what is expected. You make the single element more unstable, the entire network becomes more unstable, and the Lyapunov exponent goes up. But that's only true up to a point, and eventually the largest Lyapunov exponent goes down and even crosses zero. So over here, that's technically not a chaotic system anymore. And that is also true for the entire entropy production, which goes to zero at a finite value of this number. That's a generic behavior. And intriguingly, normally when we see in dynamic systems transition from chaos to stability, that goes along with a change of the kind of trajectory. You go from a chaotic attractor maybe to an oscillation or so. Not so here. Both in the chaotic and in the stable regime, there's an irregular state. Basically, nothing ever repeats. So the stable state actually has been termed stable chaos, and in statistical descriptions of the state sequences, of the spiking patterns, of the distribution of activity, you basically cannot tell. So dynamics and spiking statistics are really 2 independently designable aspects of biological neural circuits. So I want to just briefly show one aspect in which very strange phases happen in these spiking neural circuit models. That is a phenomenon that we call dynamic flux tubes. So what you see here is a 2D section through the phase space of this high dimensional spiking circuit model, and all these partitions are individual bases of attractions of distinct stable trajectories. When you are within such a base of attraction, there's an invariant but apparently random looking stable trajectory that the entire circuit with its 10,000s of neurons goes through. If you cross the Bayesian boundary border, these 2 trajectories will exponentially diverge on a very short time scale. And what we did in our most recent analysis that taught me a lot of statistical physics was to actually work out the dynamics of these basins of attraction, their scaling properties, and for the first time derived this intrinsic partitioning of phase space that comes from the discrete nature of interaction, that neurons feel each other only at these rare occasions when they spike, to the granularity of the stable objects with which we could eventually compute. So that's starting to raise new questions. So once we have a first handle on this, an interesting question and a key question is, of course, can we engineer these phase spaces? Can we make these bases of direction larger and smaller as it suits computation? So I'm summarizing. I want to take you to take home three things basically. The statistical physics of dynamical population coding opens a novel window on the biophysics of A to D conversion. So if we all teach our students basic biophysics, membrane biophysics, over the past 10 years, that has become an active border area between neural computation and molecular biophysics of the nerve cell. Taking that into neural circuit models as opposed to the gradual rate-like variables induces exotic phases in neural circuit dynamics. And so as one example, I briefly highlighted a novel type of a non-chaotic finite entropy phase that we found here. And I, of course, would like to thank all the people and the funding agencies that made this possible.
Fred, thank you very much for a masterful presentation. There's a lot of stuff in that. I just wonder if you might say a bit more about the sort of biological basis. I mean, why things might be the way they are. You alluded to the 3 possible explanations of which you were published, I think, in relation to the cooperative ion channel activity as opposed to loss of voltage control.
Mhm.
I mean, why are the models, what's driving, what's the biology that sits underneath the models, do you think?
Yeah, thanks for the question. These are actually three relatively intuitive mechanisms. So the cooperative one appeals to many people with a physics background. So if you, so ion channel activation happens that the open state becomes more and more stable when you raise the voltage. And that normally happens in a smooth function. So you have your Boltzmannian-like increase of the open probability. If you directly couple neighboring ion channels such that they can switch each other open, it's basically like a 2D Ising model that can have a ferromagnetic transition. So if one flips open, it can, on a very short molecular timescale, switch open the others. So that makes for a short activation timescale of the real decision makers. So the opposite hypothesis that is around emphasizes that you can, before you feed the voltage signal that is there that forms at the soma into the spike generator, you could actually high pass it. And then even a sluggish AP generator is fed by so much high pass so high frequency information that it starts to dominate. So that can be done by a huge neuron easily, because in a huge neuron, the current that impinges on the central compartment on the soma, it has to do two things. It has to depolarize the soma, but also the rest of the cell, a huge membrane area. So if you look at an input that's fluctuating very fast, actually you don't carry this large distal far away membrane area with you, and you're basically only playing with the soma and the axon. So fast things are not attenuated, but slow things can be massively attenuated in the large cell. And the mechanism is in the literature. I have a hard time explaining it because I think it's not really right, but that needs to be studied further.
Excellent, and that'll contribute to our unanswered questions ethos, if you like, of these meetings. But thank you again, and thank you for keeping to time. I would encourage everyone to think further and submit questions via the Q&A tab. We're going to take those to a panel discussion a little later on. So I'm delighted to welcome our next speaker, who is Thierry Mora. And Thierry is going to talk to us about emergent properties in a neural population code. Thierry.
So I'm going to tell you about the retina to illustrate a discussion about the link between statistical mechanics and neural populations. And the retina is a great system to ask questions, because it's a system in which we control both the stimulus. We can choose what we show to the retina, and also the output, because it's possible to record for many neurons from the output of the retina, which are the retinal ganglion cells, using multi-electrode arrays. And this response is actually all that the brain will know about the world, right? And so a natural question that you may ask, if you want to reverse engineer the system, is how does the response represent the visual information? In other words, how do ganglion cells represent visual information? That's trying to understand the input-output relationship. But maybe as physicists, we're more interested in the first, more obvious question, which is, what is the distribution of responses? And since we're thinking about a neural network, we'd like to understand what is the collective properties of this output response. It may also be the question more relevant for the brain, because the brain doesn't actually have access to the stimulus other than through this response. So maybe a first step to start is to understand what are the statistical properties. And so Bill Bialek yesterday commented already a lot about this question. So I'm going to go a bit quickly about the way we address this question. So the way we do is that we show a stimulus, natural stimulus, artificial stimulus to the retina, and we record the population response, which is in the form of spike trains. And the first thing we do is that we binarize this response to get in each time window of about 20 milliseconds, a spike word, which is the collective activity of the network at that moment, right? And now the name of the game is to try to learn the statistics, the collective statistics of this response. And in particular, we as physicists, we're interested in what are the, what are the effects of the correlative structure of that response. And so one way one can address this question is by using the principle of maximum entropy, trying to reproduce the first and second order moments of that distribution. And so we end up with solving an inverse problem, which takes the form of a familiar statistical problem, which is an inverse Ising model, disordered Ising model. And so from the correlation structure of the response here on the left, we learn an effective interaction matrix that tells us how the neurons are interacting with each other in an effective way. And once one has that kind of model, one can start playing with it, and especially from the point of view of physics. It's interesting to look at the phase diagram. One can also look for the existence of metastable states and so on and so forth. And this is something that Bilbao and collaborators have done, and something I've also worked on with him and with others. But I'm not going to talk to you about this today. Instead, I'm going to talk about, I'm going to tell you about some development that extends this kind of approach. So in a more recent work, what we did is that we used a slight variant of the Ising models, which we call the restricted Boltzmann machines. So they differ from these Ising models in two key ways. The first difference is that instead of just trying to model the statistics of the response in one time window, so the synchronous collective activity, we try to have a statistical model for the entire trace of the response, for the entire spike train, right? That's one difference. The other difference is that we're gonna now define a model in which we don't have simply the neurons speaking to each other directly, like in the Ising model, but instead we'll assume that they interact through some hidden units, which are linked to each neuron, to the activity of each neuron in each time window. And so the advantage of doing this, and this is the functional form that the model takes, we have to then, of course, marginalize over the hidden units. The advantage of doing this is that because each hidden unit here is linked to several neurons, this can actually account for higher order interactions than just pairwise. So that's one advantage. The second advantage is that each hidden unit being linked to the activity of different neurons at different times, we can also this way account for spatial temporal correlations, so cross correlations across time. So the activity of one neuron at some time with the activity of another neuron at a later time. And then there's also a technical advantage, which is that these models are actually easier to work with. It's easier to sample to sample from this distribution. It's also easier to fit to the data, right? And we actually provide online some code if you want to try it on your own data, the code to learn this kind of models. Okay, so we tested this model on retinal data from rats. And so what we show here is a randomly moving bar as a stimulus. And we checked that the model actually works in the sense that it actually reproduces the statistics of the response, of the collective response. So the first obvious thing is to check that actually reproduces the firing rates of each cell. Then we also check that it reproduces the pairwise correlations between cells. And that's important because it's actually not something that's fit directly by the model. And also the model can reproduce more global properties of the network, such as the entire population count, or the distribution of total number of spikes in the population over an extended period of time. And this is, again, a test of these temporal correlations that the model captures. And also the covariance of the different neurons across time. So now we have a perfectly working model. It's a generative model of spike trains that look very much like the spike trains that you actually see in the data. But now what do we do with it, right? And so now I'd like to go back to the question of the stimulus, right? So now let's say that we've solved that problem, trying to understand the statistics of the outputs. But what does it tell us about the input, right? So I want to go back to the input, and first I go back to the input taking the point of view of the brain itself, thinking about the task that the brain may actually want to solve, and then I will go back to a more question that maybe will interest more scientists, which is how do we learn something about the input-output relationship? So let me start with the first. The first is, can we leverage what we learned from the statistics of the output to help us in task about the stimulus? So let me introduce you to the task. So let's imagine that the retina is shown two different images. So here a bird and a random checkerboard. And we record the response, right? So if we look at, because the response is stochastic, we can do a repetition of this experiment. And if you look at two responses to the same stimulus, here the bird, for instance, we see that these responses are quite similar to each other, right? But if we compare the responses to first the bird and then the checkerboard, then the distance between these two responses would be large, right? And so in this case, the distribution of distances to responses to the same stimulus would be much shorter than the distances to different stimuli, right? So this is a case of high discrimination. By contrast, if we imagine two stimuli that are very close to each other, in that case, the responses to these two stimuli will be close by, and it would be hard to distinguish these distances, right? And so we can define a discrimination index, which is the probability that the distance of responses to the same stimulus is actually lower than the distance to two different stimuli, right? So you can see that this discrimination task is really an unsupervised task, because we're not trying to ask what is the image that you saw. Instead, the answer that the brain is asked to answer is, do you see something different, right? However, the definition of this task, I mean, to do this task, one needs to define a metric between the responses, right? And so what is the right metric? And this is where we use our modeling. So first, I will first introduce you to kind of classical way that people think about neural metrics. So let me introduce my favorite metric between spike trains. So imagine that you have two spike trains. This is just one cell, but you see two spike trains. You like to compare these two spike trains. So of course, since the spikes are often viewed as discrete events, it's pretty hard to compare these two traces, because two spikes almost never happen exactly at the same time. So the very smart trick here is to actually use to pass the spike train through a smoothing kernel, and then look at the L2 Euclidean distance between these two. And so that works for just one cell, two spike trains, one cell. But then if you have a population of cells, then the easy generalization of this is to sum over the cells, right? So I mean, this is all great. Now, the issue at least for the task that we want to define is that in a way, this distance somewhat ignores the correlated structure of the spike train. So imagine that you have two cells that encode almost the same information. So they will be very strongly correlated. In this metric, they will both contribute to the distance, and even though the information is redundant, right? So we'd like to define something that actually takes into account this correlative structure. And this is where we use our restricted Boltzmann machine modeling. So what we did is that we hypothesized that a more relevant metric should be based actually on the hidden units, right? So why do we think that's a good idea? Is because these collective units actually drive the correlative structure of different neurons together, right? So our hypothesis is that it's in this space that the metric should be defined. And so this is the mathematical definition. If you have two responses, sigma 1 and sigma 2, then the distance will be defined in terms of the expected value of the hidden variables weighted by the influence on the variable itself. And so we learn a restricted Boltzmann machine model on random stimuli. And then we did a specific, we did a dedicated experiment to solve the discrimination task. So what we did is that we chose two stimuli, so it's a randomly moving bar. So we take one trajectory of the bar, and then we ask whether just from the response we can distinguish a slightly different stimulus, so slightly different trajectory of the bar. And this slightly different stimulus has to be close enough so that the task is hard, but not too close so that the task is not too hard, right? And then we ask, what is our discrimination capability defined in this manner, right? And so what we found is that if we use classical metrics, such as the one I introduced before, but it's also true for a wide range of metrics that we used, they don't do so great. We do hardly above chance. However, if we use our metric here defined on the hidden variable, then in that case, we get a huge improvement. And that's because these, this new metric actually takes into account the correlative structure of the response. And I want to emphasize that the learning of this model is completely unsupervised, so we don't have any adjustable parameter. And again, that's because the discrimination task here is actually an unsupervised task. It's just to ask whether we see something different or not. Right? Okay, so now let me switch gear a little bit. So I've been telling you about these statistical models of the activity. And as I said, so we came back to the stimulus a little bit from the point of view of the brain asking about discrimination. But now let me go back to the question that maybe as scientists would like to answer, which is, can we basically reverse engineer the input-output relationship? But I like to do this with still using ideas from statistical mechanics. In particular, I still like to have an element of collective activity, meaning effective interactions between the cells as we do this, right? So I would like to combine these statistical mechanics models of activity with the influence and the dependence on the stimulus. So what do I mean by that exactly? So if you show a stimulus, so in this case, it's a slightly different set of experiments where we showed two randomly moving bars, the sensory neurons are responding, so they're represented by the receptive field spiking, and then you get the spikes out, right? So when we talk about generative models of the response, usually we get out the stimulus and we just have a generative model of the spikes with no relation to the stimulus, right? And there's in fact a very easy way to generalize this, which is to, in these Ising models, have the field that acts on each neuron. So it's a field that tells the neuron a propensity to be silent or to spike, to depend itself on the stimulus, and therefore depend on time. Right? So the problem with this is that learning this dependence on the stimulus is a very hard problem in itself. But what we did is that we used the trick where we decorrelate these two problems. And the trick consists in just showing a repeated stimuli so that we can actually learn we can actually learn these fields irrespective of the dependence on the stimulus and the coupling separately, right? And what we find from this is a complete interaction matrix between neurons that only takes into account the direct interaction between these neurons, right? So what do we learn from this? So as you probably know, these output cells are organized by the receptive field. So this is a representation of of these receptive fields. Here, each cell is represented by an ellipse. And what we found is that cells only communicate directly with the direct neighbors. So this is a value of the coupling as a function of the distance, and you can see that we get non-zero values only for very, very close by cells. This is at least for off cells in this network, and we also find that on and off cells don't communicate with each other. With this kind of generative model, we can show that we can indeed predict the covariance of the response once we remove the influence of the stimulus. So that's what we call the noise covariance, meaning that's the covariance that's only due to the direct interactions, and so that we can reproduce with the model. And finally, as I said, it's difficult to predict the response of individual cells even to a given stimulus, but we can try anyway. And so there's been a development in this field in the past few years. And now the state of the art for doing this is to use deep learning, so use artificial neural networks to predict the firing rate of individual cells. So we did this in the particular context of this not so simple stimulus. And now the idea is to combine this kind of model with a model of interaction between the cells, because each of these models actually are models of single cells. So we have I don't want to go into the details, but we have a way of actually integrating the two things in a somewhat smooth manner. And from that generates the prediction for the spike rates and the spikes. And so doing this, we end up with a complete generative model, which takes both the interactions between cells and the dependence on the stimulus. So to test the dependence on the stimulus, we can actually show that the model reproduces the firing rate of cells as a function of time, as we show the stimulus, with a great accuracy. And at the same time, also predicts very well the correlations between the cells for a given stimulus. And with that, I want to conclude. So, I mean, these are my conclusions. Maybe I want to insist on future work and future questions. I think that a natural development of this is to try to combine the two parts of the talk in a way, like to try to combine ideas from these restricted Boltzmann machines, which allow for high order interactions across time and neurons, with models that encapsulate the stimulus dependence. And also, I think something that I would really be excited to do in the future is to try to apply this kind of techniques to other sensory areas than the retina, such as systems we heard about yesterday, hippocampus and V1. And with that, I'd be happy to take questions. I think there are already some in the chat. So.
Thierry, thank you very much indeed for a very clear and intriguing talk. There's a question in the chat, and as you note, a couple on the Q&A. I'm actually going to use Chair's prerogative to ask you a question, which is that in terms of an application, do you think there's any, what might be the possible use of this sort of underlying models in a clinical situation? I mean, could we deduce pathology from the application of these techniques?
Well, I'm not sure I would be an expert to answer this. I think in case of pathologies, there are probably much more direct signatures that of the pathology. So here I haven't really thought about this, but I would expect, yes, that maybe the effective parameters you may learn from these kind of models would differ between healthy and non-healthy patients.
I suppose I'm thinking that given the nature of this analysis, it's possible that it might be more sensitive. A sort of preclinical index of pathology might become.
I should mention that by contrast with yesterday's talk, here we can only work with the animal models because the animal has to be sacrificed for the retina to be dissected, and so this has no direct application to humans, except that these are model animals for human health, and we haven't really looked at medical applications. I know people in the institute do, but I personally haven't really thought about these kind of problems. But yeah, I think there might be still ways to think about. I mean, this is of course an animal model, so the idea is to then bring it to humans more by analogy. And so there might be ways of leveraging this kind of technique for that.
Thank you. And thanks for the Q&A questions and the chat questions. We'll pick these up in the panel session, but also I suspect Thierry will be able to answer the two that appeared in the Q&A directly. So thanks again to Thierry, and we'll move on to our next speaker, who's Viola Presman, also from Göttingen, who's going to talk to us on phase transitions and information flow in neural systems.
So thanks a lot for inviting me. Let me start with a very innocent question. As this workshop is within the realm of physics of life, what is life? Although I think everyone has a good intuition for it, there's no clear definition. I guess like me, very many of you who are studying living systems are really intrigued by their complexity and their beauty. These living systems, they are special compared to classical equilibrium systems because they generate order, thereby they are withstanding the fundamental second law of thermodynamics. To be able to do so, they capture energy, they reproduce, they adapt to environment, they respond to stimuli, they evolve. And to be able to do all this, these living systems hinge on information processing. And therefore, I think it's perfectly the right topic to go for the study of the brain and the workshop on the brain within this beautiful framework that you have there in the UK. I would like to talk today mainly about this question of information processing in neural networks. Before I start, let me give you a rough idea of how such neural information processing might look like. Here you see the example of living computation at work. This is already a classic video. It's the entire brain of a zebrafish zebrafish larvae looked from above. Every of these little sparkling voxels represent a neuron, and the bright colors indicate its activity. Often the activity is locally confined, but sometimes you see these large waves or avalanches propagate through the entire system. Here you have about 100,000 neurons. So compared to the human brain that has about 80 billion, this is a very small system. Each of the neurons is connected to hundreds or thousands or maybe 10,000s of neurons. And the interactions are all but complying with the classical aspects of physics. So these interactions are pulse-like. These typically are spikes. A neuron is active, and then it sends a binary event to all the receiving neurons. They are directed. So if one neuron sends a signal to the other neurons, it doesn't mean that any symmetric action is traveling back. It doesn't even mean that if one neuron is connected directionally to one neuron, that there's a connection existing from that other neuron back to the sending one. The interactions are time delayed. It takes a few milliseconds from one neuron to activate the next one. And last but not least, these interactions between the neurons, they're not even fixed in time. They're changing over time. That is how we think that learning happens. These, I think, if one starts talking in this, from this interdisciplinary field of neuroscience, especially to physicists, some key features to really point out. And it is precisely because of this high-dimensional topology, so a neuron being connected to hundreds or thousands of other neurons, but a neuron does not necessarily interact with the neighboring neurons. Therefore, it is very often difficult to characterize collective properties. We can't just see them like we would see them in 2D or in 3D, but we have to have other means to try to understand their emergent properties here. In my group, we investigate neural information processing from a number of perspectives. A few topics that I want to, that we'll touch today. It's statistical physics of phase transitions. We have to develop for that a subsampling theory. We are going to some local learning rules that shape the interactions between the neurons, and looked at least a bit into the question of information theory to design computation. Let me start with the phase transitions here. It's a topic that has come up here in the workshop repeatedly from Thierry and from Bill in the first day. It is a really popular hypothesis that neural systems indeed operate close to a critical point. And if one looks into the physics of critical phenomena, this is easy to understand why that is the case. The basic properties of critical systems are well understood in the context, let's say, of equilibrium Ising model. It's a beautiful one. And I would like to very coarsely translate why the properties of a critical phenomenon might be advantages for neural information processing. So here I plotted that the susceptibility, that's a property that diverges, it goes to infinity if one changes some control parameter, here the temperature, and the susceptibility would correspond roughly to a sensitivity of a network to input. And sure, a network should be sensitive. There's more properties that diverge in the Ising model at criticality, the specific heat and the correlation length. And that would correspond, very roughly speaking, to the coding space and to long-range communication in space or in time. So this could be used, these diverging correlation lengths, they could be used for communication in space or communication in time. So criticality maximizes some abstract information processing properties. It really maximizes them. And therefore, systems at a critical point could be harnessed, and their properties could be harnessed for information processing. So it is a very popular hypothesis, and experimental evidence for that criticality hypothesis is really accumulating since the past 1 or 2 decades. There's hundreds of papers and even 2 books published on that topic. However, I really want to stress that this hypothesis is far from being undisputed. The alternative hypothesis, to just put one into the ring here, which is at least as popular, argues that the brain should operate rather in a disordered state. These people basically analyze the same data, but they base the argument on the observation that the correlation between any 2 neurons is really, really weak, comparably weak. This disordered state has its own advantages. It maximizes entropy, it minimizes redundancy, and it is connected to what we call an efficient code in the context of efficient coding. So these two hypotheses are clearly incompatible. If we would have access to the full system, we could clearly distinguish them. So why is there no solution to that yet? So to solve this problem, we had to overcome several challenges. First of all, we had to identify a common framework. We had to solve the problem far from equilibrium. And we had to, and that's the most important part, overcome the subsampling problem. So what is the subsampling problem? It is the problem that in the brain, we can only observe a few hundred or maybe a few thousand of the millions of neurons with really good temporal precision. I want to outline that here and give you a few numbers to orient you how difficult it is to assess these collective dynamics in the brain. Human brain has 80 billion neurons. A mouse still has some 80 million neurons. If we now record in an experiment with high temporal resolution, one can record the activity of hundreds, thousands, maybe even 10,000 of the neurons, but that really presents a tiny fraction of all the neurons that are out there. What we found is that if you take only a small fraction of these neurons and then make inference about the full system, then you can encounter severe subsampling biases. It might be that you misestimate the control parameter, which is very important to know what is the distance, for example, to a critical point. Scale-free properties are not scale-free anymore, and even measures as the observed correlation strength might not be representative for the full system. And these are problems that cannot be overcome. These are bias problems that cannot be overcome by longer recordings. They could only be overcome by having more of the system recorded, or alternatively, we need a closed mathematical treatment. This problem of subsampling is not only a problem in neural systems, but also in disease propagation, in protein networks and social ecological systems. Actually, our work on subsampling and propagating dynamics brought us right into COVID, which I would never have expected, but is almost a different topic. So subsampling is a ubiquitous challenge, and I want to give you now a brief glimpse on what these core problems of subsampling are and how we overcome them. In this is a simple example, we treat the propagation of activity as a branching process. This is one of the basic non-equilibrium models. The assumption here is that a single neuron is active. This could be an active neuron. It could in principle also be a person infected with a disease. And then this neuron activates a random number of neurons in the next time step. Here are the mathematical formulations. I just gloss over them very briefly. The important parameter here is the control parameter, is the branching parameter. The branching parameter defines how many neurons are activated by a single active neuron. Translated to COVID, I think that's almost easier introduction now. This is the R value, the reproduction number. If M in this framework is smaller than 1, then the system is subcritical. Any perturbation, any avalanche of activity will certainly die out. If the M is larger than 1, it means that one neuron on average activates more than one neuron in the next time step, and activity on expectation would grow exponentially. And the critical states separates this unstable from the stable regime. Now, when studying the brains, we would love to know what overall, in a mean field perspective, this very basic property is. How sensitive are these networks to input? How well do they propagate activity? This is an absolutely basic question, so to say. And without going too much into the mathematical details, if you would have access to the full system, you could relate the observed activity at a subsequent time point with the observed activity at the previous time point. If fully sampling, then the correlation between the two would be a good estimate for your control parameter, for your branching parameter. But this unfortunately only holds under full sampling. Under subsampling, as it indicated here, the correlation is not an estimator for propagation dynamics anymore. So it is, here it illustrates this idea of subsampling. If you only have a tiny fraction, if you only observe a tiny fraction of all the units, then unfortunately this straightforward and well-established estimator fails. So something as basic as the correlation strength now is, so to say, not a good estimator for propagation under subsampling anymore. And that also means that a lot of derived quantities are not representative of the full system as well. So this demonstrates that making inference about the full system by just observing a tiny fraction of the system is all but trivial. How did we overcome that problem? We have two complementary solutions. One is that we generalize from here relating activity at one time step to the next one. We generalize, oh, I should put a k here, from to the relation AT plus k. And thereby we manage to partial out the subsampling bias. So it is that for any of the correlations A and AT plus k, so k time steps or delta t time steps into the future, there's the same bias, and by using a set of this correlation, we can really partial out. An independent approach is what we call subsampling scaling. I just want to briefly mention that it's more, let's say, for the aficionados here maybe. Often criticality has been investigated by investigating these neural avalanche distributions. If the system is sufficiently close to equilibrium, then this avalanche size distribution can be used, and our theory of subsampling scaling then shows how to overcome the subsampling biases there. So going mainly or concentrating here on this estimator, this is a very powerful one for the following reasons. The subsampling estimator, it directly returns the control parameter m instead of just a binary test for or against criticality. It is very efficient, it's very precise, and it's easily applicable. Why is that? It is because you really only need to know the recorded activity at every given time step. So the number of sampled units, sampled neurons that are active at every given time step. You do not need to know the system size, the number of sampled units, that means spike sorting doesn't play too much of a role, or any of the moments of the process. Under ideal conditions, and that's from our publication, the estimation even works if you only record a single unit. Why is it? Because in a recurrent network, the single neuron basically still reads out the recurrent activity over and over. And from the temporal profile of the spiking of the single neuron, we can in principle, under idealized mean field conditions, estimate the control parameter. In real networks, so in biologically living networks, we know that we have consistent results for 150 down to about 20 neurons. Below 20 neurons, the diversity across neurons starts to really play a role. And therefore, I would say as a rule of thumb, you need roughly 20 neurons, roughly 5 minutes of recordings, and you get something like a reliable estimate of the control parameter of your neural system. This approach has already been adopted by a number of colleagues, and we have, for everyone who wants to go into that, a small toolbox out. However, the math is also not too complicated. So with this, we can go back to the slide that I introduced. There have been these, or there are in the field, these contradictory hypotheses, and they are virtually indistinguishable in experiments. We now have a possibility to apply our novel estimator and try to resolve this apparent contradiction between the hypothesis of pairwise correlations between neurons being low, leading to the hypothesis of a disordered state, or in contrast, the criticality hypothesis. And this sketch here summarizes the picture. Here on the y-axis, I put the information transfer as one of the variables. On the x-axis, it's the coupling strength, the control parameter. These properties diverge at the critical point. If one is farther away, then one is basically in a disordered state, which has the advantage of maximum entropy and minimal redundancy. The critical state has advantages of strong amplification, strong reverberations of activities, and some active memory. No one really argues for this type of a supercritical state because it has been linked to instability in epilepsy. We found for a number of recordings now in rat, in cat, in monkey, and in human, that it is operating here in a regime close to the critical point with a control parameter of 0.98. So it's neither critical nor is it disordered. We think that has distinct advantages to precisely operate in that regime. In that regime, small changes in your control parameter allow you basically to move from properties that are rather disordered to a system that is more similar to a critical state with really small changes in your coupling strength, for example, mediated by neuromodulators. So our suggestion is that this regime allows rapid tuning of computational needs of the sensitivity of a certain subnetwork to task requirements. In contrast, if you were farther away or closer to the critical point, this would obviously not work. I think I have only 1 minute left, so let me give you a 1 minute overview of what brings your system probably to this state of 0.98. What is the tuning mechanism? And there we want to dive into the difference between in vivo and in vitro activity. We know for animals in vivo that they have fluctuating activity, spiking activity over time. In vitro, however, so in a dish, the activity looks really different. It's strong bursts intermitted by pauses. And this is kind of surprising because in principle, these are the same neurons, the same synapses, plasticity principles, but the dynamics is clearly different whether it's in a awake animal or in a dish. We identified that the key difference is the input strength. These systems receive input, they're embedded in a large network of neurons. These ones are fully isolated. So we show that under homeostatic plasticity, these networks are self-organized into a critical state, whereas the homeostatic plasticity is the key feature that drives the in vivo systems somewhat away from the critical point and keeps it fairly precisely at this specific distance. Taking a model with homeostatic plasticity, just changing the input strength, we find that one can tune the network now from almost completely dissociated to this very strong bursty state, and that has major implications then in principle for in vitro systems that are being studied. It basically says that one should in principle rescue these in vitro systems from the isolation, and thereby it might be possible to establish this more in vivo-like dynamics. We also use it to tune computation in neuromorphic networks successfully. That has been very, very recently accepted in Nature Communications. So I made now a tour de force from statistical physics of phase transitions to the problem of subsampling and the solution of how to overcome the subsampling problems when assessing critical phenomena, to here a glimpse on how learning rules, in this case, this homeostatic plasticity, can stabilize these neural networks. With this, I really, really want to thank my group. This is a COVID-compatible group photo, so I'm afraid you don't see them. You only see the distance between people. And I want to thank you for your attention. Thank you.
Viola, thank you for a superb talk, which has got a lot, not just about the particular problem of neurons, but the subsampling challenge generally. And I know we'll come back to that, I'm sure, at the panel discussions. And I'm going to invite the speakers in this first session to come to the virtual podium and join the panel. And I should say that speakers from the second session will also be on the panel. So we've got a few minutes now to explore some of the questions that have been raised, and also to have a fairly open-ended discussion about this wider topic of the extent to which we are able to actually model brain behavior. And I think I'm going to ask Tom McLeish to ask a question that he's already posed in the chat, but which I know will be of interest particularly to the physicists in the audience, about this rather teasing signature of criticality versus close to critical behavior for collections of neurons. Tom.
Yes, I asked a couple, Steve, but I think it's the one about percolation.
Yes, well, you can take both, actually.
Oh, well, okay, so it seems to me that just remembering what we heard from scaling yesterday, and I asked this probably to Viola's already said something about this, but perhaps we could just kick it around a bit. There seemed to be another clear example in physics of non-equilibrium thermodynamics criticality and scaling is, of course, the family of models that go by the name of percolation theory. They have all the exponents and everything, and particularly they emerge out of connectivity. So it seems to me that there's a lot that there's some really interesting mappings one could make between the various versions of percolation theory and neural connections, particularly in regard to how close to criticality they are. So that was my question. I imagine Viola immediately has something to say, but anyone easily probably does.
Then maybe I start. So indeed, the branching process that we are investigating is of the same universality class as directed population in terms of topology or dimension. Every neuron is connected to hundreds of thousands of other neurons, so it's well beyond the critical dimension of these systems. So it's in the mean field. It has we expect, or we do see in this branching process the same exponents like in the mean field directed population. The problem is a bit that ideally we would like to have now a lot of these observables, but it is so difficult to really tackle them. All the spatial information is more or less lost because we don't know which neurons are directly connected, which are neighbors. This is why Bill Bialek then has this a bit heuristic, interesting approach of selecting those two neurons that are the most correlated ones. It's not yet clear whether that is the final solution to overcoming the problem of we just don't know which neurons are connected.
I don't know if Fred or Thierry want to comment on that same question. Perhaps Fred first.
The question of criticality is a multidimensional one, so it's hard to exhaust. So, but what I would like to mention is that in the real brain, we also have a lot of time scales in the individual elements. A single neuron already has processes in it that span from sub-milliseconds to minutes. So for instance, synapses are not fixed. That's actually important to tune a system close to a critical point. Viola and her colleagues have worked on that. But so the longest timescales of synaptic facilitation, just making a single connection between two neurons stronger, can be into the minute range. So that poses additional complexities in inferring from activity timescales that tell us about critical points. Because we are, so what we can with certainty inspect are on the hundreds of milliseconds. But we know for sure that there are single elements, biophysics timescales, that are beyond that. So I'm completely compelled that we have to follow that down, and that tuning neural circuits relative to critical points, there are critical points and they will be important. But so the step after the next step is going to be very careful about the cellular mechanisms that do that tuning.
May I comment on that, because I find it a very interesting remark, Fred. So there is obviously properties of single neurons, and then there's properties of the network, and it's an open question of how well we will be able to disentangle them. What I find interesting is the following, really again, back of the envelope estimation. If every neuron receives the input from hundreds or even thousands of neurons, and if one then assumes that the synapses that are bringing in the input are sufficiently independently changing, which may or may not be the case, then this tends to or could potentially cancel out. So that means that there's a lot of room to change every single synapse by a factor 2, 3, 4, or even more. And then nonetheless, the overall network input to the one neuron is overall fairly constant. And therefore, we think at least that it is possible to have a relatively precise tuning of a network by a few % overall, because potentially, but it's really a hypothesis that the processes within the synapses might be canceling out sufficiently. Can and might and so on. We know very little about the brain.
Thank you. Excellent. Now, I am actually going to come to Thierry for the next question, because a number of us, including Franca Davenport, had a similar question at the back of the mind. We noted that the addition of the hidden units to create the temporal restricted Boltzmann machine led to a really impressive results. But we wondered whether the hidden units had any biological significance.
Yeah, thanks for the question. It's indeed a great question. I mean, unfortunately, I think the short answer is no. I mean, but one reason is that you can think of, you could imagine them having a biological equivalent if you were imagining some purely feed-forward network. Right? So imagine you have the stimulus which has its own mode of correlations, then feeds into the photoreceptors, and then different layers of the retina, all the way down to the ganglion cells. And then these hidden units could be one of these intermediate layers. The issue with this is that the way the restricted Boltzmann machine works is that it's actually not feed forward, it's back and forth. So the visible units re-affect the hidden units and back, and the retina actually does not work that way. It's mostly purely feed forward. There's some horizontal, if you like, connections, but not really, there are no feedbacks. So but however, I mean, I do think that there is some maybe more analog, a biological equivalent more by analogy, which is that the hidden units really what they do is that they drive high order interactions between cells, right? And what could drive these high order correlations between cells? I mean, pairwise and beyond pairwise. It is, in fact, indeed, still correlations in the stimulus, also shared noise and shared stochasticity, coming from the fact that all these ganglion cells receive input from common cells, like bipolar cells, and also photoreceptors. So you could still think of these hidden units as encoding these different collective modes, right? However, you cannot really pin down exactly what is the biological object they correspond to.
Thank you. Thanks, Thierry. So a question from Alex Wade, actually, to Viola. Alex, do you want to ask your question directly?
Yeah, although, well, Viola did answer to the panel. So I was very interested in this idea that the dynamics of in vitro preparations were very sort of unlifelike, and that by activating them in some way, you could generate an in vitro preparation that was much more realistic and scientifically useful. And the question really was, how would you go about that? Do you just sort of pass current through it or something? And I think Viola answered it to some degree, but I'm really interested in how exactly you would do it. So what do you do? Multi-electrode arrays that do current injection, or what would you do there?
I think that is really, I mean, the question you're alluding to is, we know that the brain in living systems, at least typically, does not show very bursty behavior, especially if you look at cortex, it's fluctuating. We have a lot of ideas whether it's chaotic or non-chaotic, there's discussions, but it's definitely not strong bursts. And if there's strong bursts, this is indicating something like epilepsy in the living cortex. And in vitro, if you put neurons into a dish, then it's absolutely characteristic that they are silent for a couple of seconds or even minutes, and then there's a strong burst of activity, they're silent again. And there's good explanations of how this can come about. For us, one of the main reasons is that they're really isolated and that there's a lack of external input. So they have to self-generate everything. And if you go back to, for example, Michel Zodix, or what also Fred Wolf mentioned, synapses recovering and being depleted again. They might be recovering in the pause and then being depleted. So the core, at least from a theory perspective, is to save and rescue these isolated networks from the isolation. But it is still not very easy to say how to do that. And obviously optogenetic tools, they play some role. This is the way of how we are going about it. But we are about to find the optimal stimulation protocol. We are really on the way there. So in principle, I think it's 3 groups internationally that have been picking up that idea, each of them taking a bit different approaches. I'm crossing fingers and I'm interacting with them and hoping that they're getting there.
Thank you, Viola. Fred, would you like to add a comment?
Yeah, so I mean, maybe also that's an interesting instrumentation problem for people with a physics background. So Viola mentioned it, the simplest way to achieve a brain-like interfacing with in vitro neural network is to go through optogenetics. And so the molecular design community has brought, over the past 10 years, 5 years, a spectrum to choose opsins, genetically encoded actuators that fit your temporal demands of if you want to interface fast or slow and so on. There are different opsins that these people have prepared and that are quite sensitive. But that's not enough to really interface with the circuits, because if you want to feed in information in the way that a sensory stream or the input from another brain region feeds information into a local circuit in the brain, ideally, you would have single cell addressability. So that cannot be done genetically. There's not enough colors and not enough genes. That has to be done by sculpting light. So an important way is to build on holography, or on other ways to do patterned light stimulation. Holography might be really the way to go, because it's actually 3D. So a serious low in vitro model of brain circuitry, you may want to have several neurons above each other, so that's really a dense circuit. And then in order to address by optical means diffraction limited neurons in different places, holographic stimulation, fast holographic stimulation is probably the only way to go. And that comes with the last point to make. It comes with a real computational challenge, because computed holography, if you want to have different activity patterns fed into your circuits over time, that's a complex computation that we have to do every 10 milliseconds or so. So that there will be many challenges to get really to a high level of interfacing for those, even if the first good recipe has been found. So I think that's a really very interesting point, both for theorists to interface with controlled in vitro systems and for people that want to push the frontier of optical biological physics and instrumentation.
Thank you very much, Fred. So final question in this session, I just want to ask something from Francesca, because of course Francesca comes from a very large group working on this in this area at UCL, and we heard from Carl Friston yesterday, and essentially Carl was pointing out that the brain seems to operate in some sense to minimize an objective function. And he went into some really fascinating detail about what that objective function might look like. But I wondered if you wanted to just say briefly a few words about where your work might fit in that framework.
So it's not completely related in a sense, because what is working on is mostly the perception part of the problem. It is how the brain reconstructs the world and makes inference according to a given loss function. But the problem I was looking at is really how the brain makes associative learning, which is not only based on perception, it's based on a supervision signal, if you want, on a reward which is given explicitly by the word to you. You know, like you get apples, you get LT, you get cupcakes, you get not. So even if there's an analogy in the fact that my model's weights are changing by doing optimization on a loss function, which is very similar to what he's doing, the two problems are kind of complementary, if you want.
Thank you.
I could ask Francesca a tiny follow-up on that.
Yes.
I think given our interest in criticality and scaling and renormalization, Bill Bielek, after following yesterday, sent an email around saying he'd be really interested in renormalization possibilities for learning, and it seemed that you're closest to that. Can you see ways in which your methods might address a problem like that?
I'm not sure, honestly, about the renormalization part. What is clear is that so there's many people, not in neuroscience, but more in the machine learning community, who are trying to understand where this learning dynamics is going and why, right? And it's very hard mathematical problems, but there are some regimes which can be understood in details. And one of these regimes is surprisingly close to statistical physics and a renormalization, if you want. And it's also surprisingly relevant for many neuroscience problems, which is the regime I described, which is the regime where neurons, there's a lot of neurons in the brain circuits which are involved in the computation, but still, the task that these neurons are solving is relatively simple, which is the case for most of the cognitive tasks that experimentally use in neuroscience. And this is one of the limits where actually the math can be simplified. And I've tried to give you a hint about why the math simplifies. And essentially in this limit, what happens is that the networks are making very little effort to build, because they can rely on the pre-existing activity, which is rich enough to process the inputs in a way which is consistent with the task. So this also kind of links back to the concept of criticality and richness of the dynamics. So these networks are definitely using activity in the network, which is already very strong, is rich and wide, to make very little adjustments that you probably cannot even see, to make this network able to do complex computations.
Thank you.
Francesca, thank you. And again, thank you to all our speakers for a really excellent session today. We'll pick up further on the topics. I'm aware that we've really only touched on some of these, and we could talk for a long time. To those who put Q&A in the Q&A channel, you will get answers directly. And we're going to take a break, so we'll reconvene a very short break at 2:55 to 3. That's 14:55, at which point Mark Leake will take over the session. So thanks all.
Okay, welcome back. My name is Mark Leake from the University of York. I'll be chairing this, this final session in neural computations in visual and spatial cognition. I'm absolutely delighted to invite our next speaker, Remy Moneson, who comes to us from ENS in Paris, who will speak to us on from pattern completion to pattern separation using hippocampal memory encoding. Remy, over to you.
Okay, so let me first thank you for the very kind invitation to speak at this workshop here. So I'm going to tell you about some work done in collaboration with an experimental team at Institut Pasteur in Paris, led by Christophe Schmidt-Eber, and where Rui Começo-Callis actually has done most of the experimental work during his PhD. And at ENS, then the work has been done especially by Massimiliano Trippa, who is a postdoc in our group. So the work is about pattern completion versus pattern separation in the hippocampus. So we have been knowing for a long time, at least 4 decades, that actually large neural networks can act as auto-associative memories. And if I consider a network with n neurons, which is drawn here on the left, then I look at the neural activity as a point in the n-dimensional space of the firing range of each neuron, for instance, then maybe at long time, the dynamics will converge towards an attractor, which is a fixed point here. And depending on the initial condition, I may actually convert to this attractor or to another one. There might be different basin of attraction, different memories. But this is a nice illustration of the notion of pattern completion, because the initial condition within a basin of attraction is forgotten. So the kind of attractors I will be interested in today are somewhat different. There are some extension to higher dimension of this fixed point. So a fixed point can be seen as a 0-dimensional attractor, and I'm interested here in d-dimensional attractors. So this is a picture of a d-dimensional attractor in the case of d equal 1, a ring here, but you could think of planes or 3-dimensional spaces embedded in a very high dimensional n-dimensional space of neural activity. So here the notion is that depending on the initial configuration, then a fixed point along this attractor will be reached, and then in the absence of noise, then the dynamics won't change, and this neural configuration will be frozen. Starting from a different initial configuration, I may end up here. So why is it interesting to have d-dimensional attractors? Well, it's interesting if the memory item, the memory actually you would like to build, has to do with a continuous variable, because the coordinate along the attractor, which I will call x, which is a collective coordinate extracting from the n-dimensional neural activity of a population of neurons, can actually take any continuous value along this d-dimensional attractor. And there are many situations where actually this kind of mechanism for building memories of continuous variable can be useful. So one example, which has attracted a lot of attention over the past decades, are place cells in the hippocampus. So Bill already told us a little bit about place cells yesterday. So let me just, as a reminder, explain again what place cells are. So place cells are present in the hippocampus regions CA1 and CA3, and they essentially have spatially localized firing field. So what does that mean? If you take an animal in an environment, which is a room here, as the animal is moving, actually, the activity of the cells in the hippocampus will change depending on the position of the animal. For instance, this green cell here, this cell will be active when the animal is around this spot. That defines a region of activity, which is called the place field of the cell. Another cell will be active somewhere else here and has a different place field. So place cells actually and place field enjoy a lot of very interesting properties. They are stable in dark and against limited changes of the environment. So they are not only driven by visual stimuli, they could actually depend also on other kind of sensory information like olfactory information. The place field also code for they could depend on the emotional state of the animal. It could depend also on the task that the animal should perform in its environment. And they are very stable in the sense that when the animal learns for the first time in an environment, then after a few minutes, then the place fields are formed and they can be retrieved in the same environment after days. So one very crucial property is that actually place cells fire, as I said, as the animal is undergoing some physical motion in the trajectory in the environment, but you can see also sequences of activation of place cells when the animal is sleeping or immobile. So in that case, the sequences of activation can take place on very much compressed time scales, which shows that it's not only a property of some kind of stimuli which is driving the activity of the cells, but there is really an activity of an underlying network which is producing these sequences of activation. So what could be this network? So first model was proposed by Sodik and Sejnowski about 25 years ago. And the way it is built is actually quite simple to explain. So imagine you have n cells, and I call Ri the firing rate of cell number I. Then the first equation here is a first order differential equation for the change in time of the firing rate of cell I. You get some leaking term here, which would actually drive back the firing rate to rest value. But then there are also some inputs coming from currents. So G is here a nonlinear function, which tells you how the activities of the other cells actually will contribute to the firing rate, or to the increase or decrease in the firing rate. So the Jij coefficients, which are the synaptic coefficients here, are given by this formula, which is the crucial one here. So each neuron I is associated to a spot in physical space, xi, which is the center of its place field. And you will have, with this formula, that two cells I and J whose place field centers are close to each other will have a positive, strong coupling, while two cells which are far away, so that this term is actually very small, will have a weak negative inhibitory, inhibitory coupling. So the mixture of local excitation and global inhibition produces something that we well know in physics, like for instance in the gas vapor in the gas liquid transition. So we'll have a droplet or a bump of very high density here in a liquid, or let's say a bump of very active neurons with high firing rates, which is surrounded by a low density vapor, or let's say a lot of neurons with very low activity. And here is what I shown here is just this bump of activity where each neuron is located here. So you see a firing rate of neuron I located at the center of the place field xi. So these bumps define a collective coordinate of the activity. It can move in the plane, and as it moves, actually, its center of mass codes for the position of the animal. So the important thing is that the existence of a bump is due only to the pattern of recurrent connection here, the Jij matrix, but it can be driven by a very weak input, which is, for instance, a stimulus. And in the case of the hippocampus, in the CA3 region, which is actually known to have a lot of recurrent connections, then this external stimulus, which depends on the input, which depends on the position of the animal, could actually come from the medial and terminal cortex, which is a very interesting region where cells are found with very peculiar properties, such as grid cells. And I think Alessandro, in his talk right after me, will talk about these grid cells at length. Okay, so you see, these inputs actually can actually drive the bump, which is due to the recurrent connection here, and then the bump will move along this attractor in the n-dimensional space. So, of course, we can actually, or an animal can memorize many representations of in the same hippocampal network. each representation corresponding to a spatial map, a particular environment. So far, people have done experiments where they have seen that 11 maps or 11 rooms were actually encoded in the same animal, and they were able to measure these maps. So that means that the picture is more similar to what is on the right of my slide here. You have different attractors, and depending on the particular initial condition, where, for instance, the animal will be actually moving its neural representation along one of the attractors which correspond to the room it is in. So there are plenty of interesting questions about how many attractors you can store, and what is the accuracy of attractor, spatial resolution of the coding position, and so on. What is interesting also is actually to think about the dynamics to jump from one attractor to another one. And here you can think, for instance, of the same environment where an animal can actually look for food in quiet conditions, or try to escape a predator and go to the shelter in more complicated times. And you see that the same environment should be actually represented by completely different representation depending on the external context, and the representation should be able to actually flip from one state to another one very quickly, and that has been seen in experiments, in particular done by Fenton and collaborators. So in my picture of attractors, the question is how you can jump from one attractor to another one d-dimensional manifold to another one. And this we have studied by looking at the dynamics given the networks. Can you actually understand the scenario of this jumping dynamics? But you can do also the opposite, and this is related to what Bill explained yesterday, and also Thierry in his talk today. Can we actually, if we had some experimental data about neural configuration, can we infer the connection of a network and actually see how this network is able to jump from one attractor to another one, and interpret the representation that is in the data, which is encoded in the data. So what I want to talk today about is actually related to this question. So I want to talk about dynamics, but jumping from one attractor to another attractor, but in a very peculiar case, which is actually quite important, which is a case where one attractor is well formed because it corresponds to an environment that has been learned by the animal over time, and the other one is actually not well formed. It's some kind of pre-map or pre-attractor, which is somewhere in the limbo, but which is not consolidated at all. So I'm going to back to this question in a few slides. So this is an issue which is actually crucial for pattern separation, which is actually the other kind of task that the hippocampus has to do. So when you enter in an environment that you know of, then the hippocampus should be able to retrieve the map that you have learned in the past experience. But if, for instance, a piece of furniture has been changed in the apartment, it could be its place has been changed. It could be that actually this change of location has a very important meaning and should actually trigger some decision and some action. So maybe in that case, a different representation should be attached to this new position of a chair, of a table, or whatever. So so the first task is pattern completion. The second one is pattern separation. How can both tasks be actually performed and carried out by the hippocampus? So, of course, after a long time, when a new attractor can be consolidated by adding maybe some new synaptic connections or reinforcing some connection, maybe weakening other ones, and then we'll get back to the situation I was mentioning before of two competing attractors. But here I'm really interested in this talk about this situation when we have a consolidated one and a pre-one. Okay, so the experimental setup that our collaborators at Institut Pasteur were considering is actually the one which is shown in this picture here on the left. So a mouse is actually experiencing some virtual reality situation where a movie is projecting onto a wide-angle screen, and the mouse can actually run the movie by rolling the ball here. So the mouse is immobile, but it can, with its leg, roll the ball, and the movie is seen on the screen. So there will be two types of movies. They are very similar in terms of pictures which are proposed to the animal. So the first one will be the familiar. So the familiar movie that you see here, this is just one picture at one time. So it's a 1D corridor, and the mouse is virtually moving along this corridor, and the movie is playing as the mouse is running the ball until it reaches the end of the corridor. At that time, the mouse is transported back at the departure position along the corridor and so on. So there's several trials, one after the other. And there will be also movies which are very similar but which are different. They are called novel movies. The difference is that the familiar movie is played many times, while the novel movie is played just a few times. So the animal was not trained with the novel movie. So what is the task the animal has to do? It has to move along this track and find a spot, which is a spot where if it pauses, then the animal will be rewarded with some water. If it doesn't halt, then there will be no reward. And so the animal is, let's say, punished because it has not understood that it should stop there. So the experiment is done in this way. So for 5 days, the animal is trained in the familiar movie, and the hit rate increases. That means that the animal understands better and better that it should stop somewhere in a specific spot here to get the water. And then on day 6, then the novel environment is presented. And what you can see, it's presented in an interleaved way with the familiar one. So you can see when the familiar is presented again, then the animal has a very high probability of finding the reward zone, while with the novel one, it cannot, obviously, because it was not trained. So the question is how actually, if we go on with the experiment, eventually the animal will be able to be trained and to learn also where is the reward zone in the new environment. But the question is what happens right at the beginning when this novel environment is presented. Okay, so as I say, the hippocampus is actually receiving a lot of inputs from other cortical areas, in particular from the medial and terminal cortex. But it also is coupled to other regions, and one region which has been known for a long time to be crucial in pattern separation is called the dentate gyrus. So the dentate gyrus is very peculiar because it has many more neurons than the hippocampus, about 1 order of magnitude more, but most of these neurons are actually very much silent. So it's very, it's characterized by very sparse firing, about maybe 3 to 4% of the neurons are active, and a huge expansion. So this very particular architecture actually has drawn a lot of attention over the past decades. And Edmund Ross and Alessandro Traves were among the first ones to understand that and to suggest that dentate gyrus could actually be very important for pattern separation. So, so over the past years, a lot of experiments have been done on the dentate gyrus. So that used to be very difficult to record activity due to the sparsity of this activity. But now with optical recording, this is easier to record the activity of large regions, as we heard of yesterday. And it's known also that animals with impaired dentate gyruses, for instance, are unable to distinguish two environments which are close to each other, which are similar, but for which two different tasks or two different behaviors are expected from the animal. Okay, so what we and Christoph did is they first record the activity with whole cell patch clamp techniques of granule cells in the DG, in the dentate gyrus. So you see one of these granule cells here with the scale, the size of this cell. And what you can do, and what they did, is that they injected some current in the cell and they could measure the membrane potential. So here you see the current, for instance. So you see 4 different currents which are applied for about 200 milliseconds, and you see the response of the cell. And this is the difference of potential between the inner and the outer part of the membrane. So if the current is weak, the cell is barely changing its value of the potential, which is about minus 70 millivolts. That's the rest value. But if the current is actually able to drive the difference of potential above a threshold value, about minus 45 millivolt, then the cell is able to trigger a lot of action potential spikes, and you see that for different, for this interval of time. So the question is, what happens now if you don't do this driving with external current, but just measure in vivo the membrane potential V as the animal is moving virtually in the maze, in the environment. So this is what I'm showing here on this slide. So let's look at the, let's say, middle panel here. So the x-axis is time. The time scale is about 10 seconds. You see that here. And the y-axis is the position in centimeter in the virtual environment. So this is the position which is shown by the movie as a function of time. So you see different trials here when the animal is actually running from one extremity to the other one in the familiar environment. You see 3 of them with essentially constant speed. And then after some time, you see the novel environment which is introduced. So the movie is changed, and there are 5 trials here, and then it's back to the familiar one when the speed is actually lower. So if you record the membrane potential of one cell in the DG, you get this kind of time trace, which is at the bottom of the slide here. So as I said, most of the neurons are silent in the DG, in the dentate gyrus, and this is one of them. If you pick up with one electrode randomly one of the neurons, the odds are very high that this is a silent neuron, and in fact it is. You see some fluctuation around the rest value, which is around -70 millivolt. Actually, if we look more closely to this trace of a potential, something happens. So here, the vertical red bar corresponds to the time of teleportation. So you can teleport the animal from familiar to familiar environments when it reaches the end, and then you put it back at the start. Or you have also teleportation from familiar to novel. So the gray trace is a familiar to familiar teletransportation. And you see essentially that the average value of the potential as a function of time, and this is average over many trials, is fixed. It doesn't change. Nothing happens. In the familiar to novel transition, what you see is a short increase for about 1 to 2 seconds of the potential. So this increase is about 1 millivolt. So this increase is very reproducible. It's found only when the animal is engaged in a behavioral task. It has something to do. If the animal is immobile and there is no reward, that you don't see. And it can be actually erased by blockade of some neurotransmitters which are known to be very relevant in attentional processes. So it seems that 1 millivolt is actually not very important because there is a long way to go from -70 to -45 millivolt in order for the neuron to be active. But actually 1 millivolt, we should care about this 1 millivolt because for the following reason. If we look at the histogram here of the average values of the values of the potential over time for the neuron, you see that this is centered around the -70 millivolt rest value, and sometimes there is a small tail here which is barely visible, where it crosses actually the -45 millivolt threshold, and then the neuron becomes active. Now, it turns out that this occupies only about 3% of the time, so it's very weak, but by shifting the whole distribution by 1 millivolt to the right, then you can go from 3 to 4%. So that means that relatively to the baseline activity, you can see an increase in activity, which is about 25%. So 1 millivolt actually has a huge effect on the activity of the DG. And this effect is about 1 to 2 seconds long. So the question is this effect sufficient to do something to trigger some change of activity in the hippocampus? So this is where Massimiliano actually introduced a computational model. So the model is actually...
Remy, apologies. Could you finish within about 1 minute, please?
Yes. So the model is the following. So you have a consolidated map, which is very similar to the continuous attractor I was mentioning before. And you have a map which is actually highly degraded with very few connections. And if we actually combine, here I'm showing what is going on, if we combine an inhibition, transient inhibition of the hippocampus, which is known from other experiments, to shut down the map in the familiar environment, and we excite randomly cells all over the place in the hippocampus due to this transient increase of activity coming from the DG, then we are able to evoke a very ugly bump of activity in the new map, but that's sufficient for some time. And the other map in the familiar environment is completely shut down. So this is summarized in my last slide. So if we have a nice map here for a well-known familiar environment, then we have this continuous attractor where the MEC, actually the medial internal cortex, can drive the activity along the maze. If we shut down this activity with inhibition, then actually and stimulate other neurons with the transient activity of the DG, we can switch on a bump, a partial bump, which can be maybe later on consolidated. Okay, so I think I should finish here.
Okay, many thanks, Remy. That's a fantastic work in modeling pattern completion and separation. We're running a little bit over, so we won't take any questions now, but attendees, please do post your questions in Q&A, and hopefully we can either come to those in the panel discussion, or Remy can address them directly. So straight on to our next speaker. It's my pleasure to introduce Alessandro Treves from SISS Trieste. Alessandro will speak to us on grid cells embrace disorder and turn into an ultrametric glass. Alessandro, many thanks.
So thank you, everybody, and the organizers, and also thanks, Remy, for a very nice talk, to which mine is a kind of follow-up. So I want to talk about grid cells. Those of you who have who don't know about grid cells are a bit like base cells, but with each cell has multiple places, multiple regions of the environment in which the animal, typically a rodent, moves around, which make a particular cell active. So here at the bottom, you see 4 examples of grid cells, and you see that each has multiple regions of activity, multiple place fields. And in addition, there is this striking feature that for a given cell, the fields of activity appear to sit at the vertices of a regular triangular grid. We can see a particularly nice example here. And these were cells in the region that is just before the hippocampus in information flow, the medial entorhinal cortex that Remy has mentioned, discovered by Edward and Maybrit Moser and collaborators, and that led to 1/2 of the Nobel Prize in Medicine and Physiology in 2014. So there is this beautiful phenomenon that suggests that somehow in one part of the rodent brain, deep inside, there is a spontaneous emergence of mental order. It's I stress order that is not in the environment. This grid is not in the environment. It is in the cells, and it is in each cell a different grid. So each cell expresses its own grid with local coherence among cells that are nearby in the tissue. A surprising property is that if you take this rat and put it in a different environment, you see that the cell you're recording from expresses the very same grid. So in some sense, at the level of individual cells, there is something like a universal metric expressed by that cell. This is beautiful, but also as a kind of strong implication that this grid cell system probably has no memory. If it expresses always the same metric, that suggests that it cannot distinguish between, for example, different environments. And this has been quantified already many years ago in work by Marianne Finn and Torkel Hafting in the Moser lab, in which they could show that whereas in place cells, there is the information, the red dots, required to distinguish an environment A from an environment B, relative to distinguishing A from itself. In grid cells, there is no such information. That kind of resolved the long-standing tension between 2 different accounts of what the hippocampus does. The hippocampus with the CA3 region where you find the place cells, the account that would explain it in terms of a spatial function, and the account that would explain it as a memory device, championed by Brenda Milner, an amazing neuroscientist still alive at 102 years of age. So the discovery of the grid cells with their apparent universality means that the hippocampus is the only place where you can have memory. So it suggests that the role of the entorhinal cortex has not to do with memory, but to provide the hippocampus with some of the spatial information that goes into memory representations. Okay, so there is this universality, at least that's an observation in the experiments that have been done, but one could ask whether in principle, the grid cell system, if it is implemented by a recurrent network, could it have distinct attractors like Remy has just mentioned in his talk? And we could address this in principle in a mathematical model. We have done that with in collaboration with Remy and his collaborators. In fact, we have analyzed two different mathematical models that I don't describe in detail that produce from very different perspectives, essentially converging results. The results are surprising, that if you were to store in these recurrent networks square maps, the storage capacity would be so low that in practice, in a realistic network, you could store just one such square map with a square grid. But the triangular, or we call them hexagonal grids that are seen in the grid cells observed in rodents, the storage capacity is much higher, is of the order with realistic parameter of 100 or so different maps. This is a kind of simple simulation by Davide Spalla in which you see in simultaneously one map and another map. Only one map is retrieved and leads to this localized bump of activity. So grid-like units can in principle express different orthogonal metric representation, maybe of the order of 100 different environments. But the question is, will they do that? And it's a question to which recent experimental observation have indicated the answer is non-trivial. And one of the most interesting recent experiment has been to try to look for the activity of grid cells or would-be grid cells in 3D space. Not in rodents, but in bats, mammals that fly and that can sample more or less homogeneously, at least some volume of 3D space. So if one were to observe something regular, like the grid cell we see on a 2D environment in 3D space, one would expect to see something that looks like a crystal, like this structure that I put up, which is purely a theoretical expectation. A theoretical expectation that has not been met by experiment. And I cannot show you the experimental observation, they're not published yet, but I show you the result of simulating our model for the emergence of grid cell, which is still a model, but produces these kind of disorderly fields in 3D, which in fact are pretty much a description of what Nahum Ulanovsky and his collaborators find at the Weizmann Institute, and it's part of the reason the paper is not yet published after several years. It's a beautiful experiment that unfortunately does not produce a beautiful crystal-like organization of the selectivity of these cells in 3D. So this is an indication that what we see in 2D environments may be something that we over-idealize. Another indication for that comes from other experiments in which they've put rodents in environments with odd shapes, like this American college flag, in which, if you look closely, you find that the triangular grid is a bit irregular. It is as if broken at some point. And the same has been found in large environments by the Moser group, in which, again, you see a kind of broken long-range order. So these grid cells are not really quite like a crystal. And then at this point, it's good to think again what we have been doing and consider that we have always been doing these experiments. People have been doing these experiments in very artificial conditions, placing rats in these flat, empty, square boxes, often square boxes. A bit like if we were to look at humans only in these very artificial environments that are very much unlike the natural environments in which we can presume our nervous system has evolved. Natural environments are usually far from flat and square. In Jerusalem, the flat square box has been put by humans after a landfill, but originally the environment was with valleys and hills. And you can ask whether a grid cell representation on such a non-flat environment, what it would look like. We don't know yet from experiments, but we have tried applying our model to at least regions of constant positive curvature, like a sphere, or constant negative curvature, like a so-called pseudosphere. And the answer is quite simple, that you can have regular grid cells, but they have non-hexagonal symmetry. They can have pentagonal or lower symmetry, or heptagonal or higher symmetry, depending on whether the curvature is positive or the Gaussian curvature is positive or negative. Now, this has important implication. The deviations from six-fold symmetry have important implications on the coherence that you can have among different grid units. If you run a simulations on a sphere, for example, the emergence of a grid cell on a sphere, you find that different grid units, would-be grid units, can evolve a selectivity with, say, 12 fields, like the perfectly symmetric soccer ball, or 13 fields or other numbers. So immediately realize that cannot be complete coherence among units that have different number of fields on the same sphere. But even among units like these two units that are pretty close to perfect soccer balls that have 12 fields, they cannot display coherence under translation, simply because translations are not possible on a sphere. So that means that there cannot be long-range coherence, long-range in the sense over the whole sphere, between units that, even if they express perfect symmetric grids on a sphere. And the same is true on any curved surface. So coherence in the sense of translational coherence, what we see in experiments of grids with grid cells in flat empty environment, is only possible on flat empty environments, which are unlike real environments of rodents, or here we see a particularly fancy environment of a kangaroo rat, which is curved everywhere. But even in a standard Norway rat, where people, with which people do experiments, is a curly environment in which, if you take as a parameter the ratio of grid spacing to the radius of curvature, there is a very limited portion of the range spanned by a realistic environment where the curvature is so small that the environment is effectively flat. And only in this region there is this six-fold symmetry. Outside, there is a coordination number above or below 6. So it's a bit like a situation of the connectivity in our brain, that only in the corpus callosum, in the middle between the two hemispheres, there is coherence in the direction taken by the different paths. Okay, so I will report next time about what happens in a realistic environment. But now I just give you, in the last 1 minute or so, some snippet of what happens on a sphere, which is an interesting model because it's a model which people have used in experiments, both experiments like Bill has mentioned of the tank lab, or also Remy has mentioned, in which a rodent walks on top of a sphere or of a revolving sphere. And also you can have experiment where a rodent is inside the sphere. The Gaussian curvature is the same in both cases. So we can look at what a model does on a sphere. And one first key result is that if you have grid cells that do not interact with each other, with no collaterals, they can approximate a perfectly symmetric grid unit. But the moment they interact with collaterals, they become much noisier, simply because there is this impossibility to form a coherent network. This is work by Federico Stella, which points at the fact that collaterals make coherence impossible. They slow down the dynamics of the network, how the cells develop their selectivity in time, something that reminds us of some slow glassy dynamics. And different cells unable to form a single coherent network break down into clusters that to some extent remain also when the animal is moved to a different environment. So clusters that survive remapping. I don't have time to give you the full results, but I just want to show you this last result. We find that for grid units that evolve their selectivity on a sphere, they form these clusters. The degree of coherence they express is kind of exactly intermediate between the full coherence that you could have, say, on a plane, and the completely uncorrelated selectivity that you would have with units that do not interact. In terms of this scaling with respect to a parameter that I'm not explaining to you, instead of having a 1/n scaling, you have a 1/√n, something that we think is a kind of interesting physics of this system that we still have to fully understand. So I conclude with a kind of take-home message that the implication of these beautiful grid patterns that have been discovered on a flat, empty environments could be quite different once we translate the phenomenon to the realistic environments where rodents and other species have evolved their nervous system. So we should not derive too fast conclusions from this beautiful long-range order that we see in the experiments in the lab. Thank you very much.
Alessandro, thank you very much. Fantastic exposition of grid-related modeling. I think we've got time for maybe a couple of questions. I'm going to read one out for you. It's from a Mashir Harsh. And the question is, well, it's a multi-themed question. How are these grids related? Is there some transformation between them? Same lattice length in all, and question mark orientation? What do you say?
Very good questions. And the simple answer is that grid units that are close to each other in the tissue, they share the same spacing and the same orientation, but not the same spatial phase. So their fields are not in the same place, but they're like shifted one to respect to the other. Whereas if you go longer distance in the tissue, as you go deeper from the more dorsal to the more ventral part of the rodent brain, you go to regions where the spacing is typically larger and larger. In fact, in the Moser lab, they've discovered that this is not a gradual increase. There are what they've called modules. In each animal, they find between, say, 5, 6 different modules where the spacing in each module is relatively constant, the same. We think that this is not a kind of genetically instructed organization, but it's a phenomenon of self-organization.
Very good, thank you. I have finally what is probably a very dumb technical question, but how important are non-nearest neighbor interactions in your modeling?
So basically, these units have, like pyramidal cells in general in cortical systems, they have very high connectivity. So we should not think in terms of physical systems that live in 2D or 3D, even though the cortex is a kind of two-dimensional tissue in some sense. but the connectivity between the units is very high. Still, there are phenomena that reflect the kind of the neighborliness of different units, and the modules is one such phenomenon, that units that are closer by in the tissue have more connections, they presumably share more connections, and therefore they tend to express grids with the same spacing. Yeah.
Very good, thank you. We have at least one more question in the Q&A. I think best if you can respond to that directly in the Q&A, and we can also try to follow through in the discussion panel there. So Alessandra, many thanks again, fantastic talk. And now we will move on. Okay, so our next speaker is Alex Wade from the University of York. Great pleasure to introduce Alex. He is going to talk to us on altered visual processing. Good. Well, you can see the title there. Fantastic. Over to you, Alex.
Thanks. I'm not sure this is the title. And in the spirit of the physics theme, I think what you're about to experience here is an impedance mismatch between the beautiful mathematically precise talks that we've had this afternoon and this, and I don't think you're going to be struggling over the mathematics of this talk, which is really for everyone. So just as we start, I'd just like to thank all these people and more who've contributed to this research, and I'm going to go quickly, but I'd particularly draw attention to Chris Elliott, who's been unfailingly generous, both with his intellectual input and with his lab resources over the last 10 years that we've been collaborating. It's just been a really wonderful person to work with over the last decade. So as part of my life, I'm in this business. I'm in the business of trying to understand what's going wrong with the human brain during neurological disease and how we can maybe make therapies that make people better. And I think you can characterize drug development in this manner, that people try to give animal models phenotypes that match human diseases, and then they try to develop some sort of therapy for those animals, and then bring the therapy back into humans. And what I'm going to spend the next 15-20 minutes trying to convince you is that this is a perfectly good alternative to the mouse, and that it comes with a number of really important benefits that I think we should all just be aware of, and that I love flies. Flies, you all know the fruit fly, Drosophila melanogaster, maybe from biology at high school or in first year of university. But there's a huge conservation of machinery between you and a fly. So you last shared a common ancestor with a fly maybe 600 million years ago, but it turns out that's not that long in evolutionary time. Many of the genes are conserved between you and flies, many of the proteins, many of the biological processes. An interesting rule of thumb, 70% of human disease-causing genes have fly homologs. Drosophila scientists can take apart the fly genetics and put it back together almost at will. So compared to mice, flies are uniquely manipulable at the genetic level. You can put things in and out of them, and they display complex behavior. So they're little animals. They do things. They live and they love and they reproduce and they learn and they have memory. And finally, they reproduce so quickly that you can generate new generations within about 2 weeks. You can have a whole new generation, and there are thousands of them. And also, interestingly, they're not subject to ethical control. So of all the animals you might want to use in a lab, even fish, even the zebrafish are subject, because they're vertebrates, are subject to some ethics controls, but flies are not, because they're invertebrates. So if we want to use flies to study neurological disease, we're interested in how we can get information in and out of them. And it's a fact that many human neurological diseases now appear to have visual correlates. So people with these diseases report changes in their vision. And this is where we're going with the flies. And we'd like to understand something about the similarity between fly vision and human vision. So the fly visual system and your visual system evolved separately. The visual system of the common ancestor was probably not really an eye. Nevertheless, it was noted quite early on by Romani Cajal that flies and humans, if you look at them at the sort of the cellular level, appear to have undergone a convergent, parallel evolution, where features such as, for example, the bipolar cells in humans, bipolar cells in humans here are mirrored by a sort of similar set of cells in flies, the photoreceptors here, and so forth. And Ramon y Cajal even went so far as to interpolate between the two species with this flirtebrate, where he sort of explicitly drew analogies between the different processing stages of the different animals. So if we want to record population responses from awake, uncooperative small animals, we need a technology to do that. Unfortunately, here's an awake, uncooperative small animal being recorded from, I hadn't noticed until now that he's wearing, or he or she is wearing a USC little baby grow with the little USC mascot on it. So we know where this picture was taken. So this child is watching a screen on which a flickering pattern is being presented. And what they're doing here with this child is measuring their contrast response function, or the way that their brain responds to visual inputs. And the technology here was described by Dan Baker yesterday, one of my collaborators. If you flicker a stimulus on the screen and record the population responses through an EEG cap, what you get out of the brain is a phase-locked, time-locked, frequency-locked response that perfectly reflects the input that's coming in. And by measuring how particular inputs of these frequencies drive responses in the brain, you can build up an input-output relationship between the population neural response in the brain and the input. So to do this with flies, we had to translate the technology down to the level of a fly. We had to build a fly TV, and it's a rather nice illustration of what we did. In reality, one of the facts about flies is that they're just very small. So this is one of the standard yellow Gilson pipettes, and the fly is inside this pipette right at the very end. And in this rig, we're recording using a single electrode from the fly's eye, from an electrode placed directly onto the fly's eye. So flies are very small. You can place them inside these Gilson pipettes and stick them there with a bit of nail varnish. And then you can bring an electrode into contact with its mouth and into contact with its eye and record an EEG response from its head in exactly the same way that you might record an EEG response from a child's head. And if you pulse a flash of light, for example, on the screen, you'll get this very stereotypical VEP from the fly's eye. So in the world of steady state, we present flickering stimuli that drive periodic responses in the visual system. So here you're seeing the responses to two different frequencies, which can, by the way, be presented in the same stimulus. So the first frequency here is in red, and the second frequency, the first frequency is in red and the second is in blue. And you can pick these apart using a Fourier transform of the time domain response. Here you can see a different set of responses. These are from two different flies. This fly here has a strong response at the first harmonic of the input frequency, a relatively weak response at the second harmonic. In this second fly, that pattern has been reversed. We can also do a cute thing that vision scientists have been doing for almost half a century now, which is examine the responses to one particular contrast probe in the presence of another constant contrast input called the mask. So for example, we can sweep the contrast of one input at one frequency. We can have another input at a different frequency remain at a constant contrast, and we can see what the effect of adding or removing that mask is on the input contrast response function. The reason to do this is to examine a phenomenon called gain control contrast normalization, where the responses, the visual system changes its gain depending on the ambient contrast of the scene. And by adding and removing this mask, you can generate a sort of very stereotypical leftward or rightward shift of the contrast response function. This was first noted by A.B. Bonds in '89, and we now, thanks to work by, for example, Matteo Carandini and David Higa, have a very mathematically clear description of what's going on in these computations in the early visual system. So here's work we did over 10 years ago now, looking at the similarities in these response profiles between humans, and in this case, we looked at cats as well, and found an almost identical change in the response pattern in humans and cats as we probed the contrast of the flickering stimulus up over a range of contrast, and then added or removed a mask. So adding a mask changes the sensitivity of the neuronal population. It shifts these responses to the right. You can also look at the responses to the mask, and it itself is crushed by the presence of the probe input at a particular contrast level, and exhibits this very stereotypical nonlinear decay. When we do this same experiment with flies, just to reassure ourselves that we're getting something sane, so here's illustrating the rightward shift of the contrast response function in the presence of a mask, and the crushing of the mask response in the presence of the probe. Flies exhibit almost exactly the same response. So here you can see this contrast response function in the fly, EEG shifting to the right, and the crushing of the mask input in the presence of increasing probe contrast, this very sort of nonlinear response. So we're interested in neurological disease, and the neurological disease that was being investigated by Chris Elliott's lab, I think fortuitously at the time, was Parkinson's disease. So it's been over 200 years now since James Parkinson described the neurological disease that now bears his name. And recently it's been shown unequivocally that as well as the traditional symptoms of, for example, tremor and hunched posture and akinesia, many Parkinson's patients report a sort of an array of hard to pin down visual disturbances. We're particularly interested in genetic forms of Parkinson's disease because we can use these forms we can place these genes into our flies. And the form that we're looking at is the most common form of genetic Parkinson's disease, a form which is caused by a mutation, G2019S mutation, in the kinase domain of the human LRRK2 gene. So this leucine-rich repeat kinase gene in humans, it has a relatively unknown function. I think it's starting to be understood over the last 10 to 15 years. But this mutation here in the gene in the kinase domain leads to constitutively increased kinase activity in this gene and will lead in about 50% of carriers to Parkinson's disease. Probably the most famous carrier of this disease at the moment is Sergey Brin, one of the co-founders of Google. And it was known 10 years ago in Chris Elliott's lab that if you place this gene as a transgene, this mutation as a transgene into flies, not only do they express it, but the presence of the mutation leads to loss of vision in older flies. So in relatively young flies carrying this mutation, you see relatively little effect on the visually evoked response, but by 28 days, these are very senior citizen flies, they've gone essentially blind. And this effect does not happen when they simply express the normal unmutated human transgene, nor does it happen just in normal, relatively wild-type flies. So there's something about carrying this G2019S kinase mutation in flies that leads to a loss of vision. And it was hypothesized at the time that this was an activity-dependent decay, that somehow this increased kinase activity would lead to increased activity early in the fly's life or an increased sensitivity to light, and that would generate an excitotoxic cascade, which would lead to cell death in later life. So when we came in with these steady state VEP techniques, which are exquisitely sensitive to changes in the amplitude and also the phase of the signal you get out of the visual system, we were able to make much more precise measurements of what's happening in these fly brains as they receive visual input. And so here are the responses of wild type flies. And these red lines are showing the unmasked response, and the gray lines are showing the unmasked response. The red lines are showing the masked responses. And in the presence of a mask, you can see that the response is slightly decreased. These are the different harmonics which represent activity from different layers of the fly visual system. So we think that the first harmonic of the response originates primarily in the photoreceptor layer. The second harmonic is originating in the first neurons in the fly visual system. And this intermodulation term, this beating between the two inputs, is arising deeper in the fly brain. So neither wild-type flies nor flies carrying the human unmutated transgene show any unusual responses, but in the presence of the G2019S mutation, you can see that there's massively increased excitation in these flies. So the response, even at the photoreceptor level, is increased. At the second neuronal level is massively increased, and we see this significant change in the way that gain control operates as well. And this peak here at the intermodulation term is much more pronounced, suggesting that, again, something to do with gain control is altered in these animals. This kinase domain is a target for drug companies, so the logic being that if you could shut down the excess activity in this kinase domain, you could somehow cure Parkinson's disease. And so it's the target for many so-called kinase inhibitors. This box has moved across, unfortunately, but this should be on the kinase domain. But the idea is that you find molecules that bind to the kinase domain and shut it down. And these kinase inhibitors are drug candidates for Parkinson's disease. We were very generously donated two of these candidate kinase inhibitors, one by Lundbeck and one by a group up in Dundee who make it their business to make these types of things. And we showed using this steady state VEP technique that, so again, here are the wild type flies exhibiting this normal contrast response function. Here are the mutated flies, or the flies carrying the human transgene, showing this massively increased response, this hypersensitivity to visual input early in their life. And then here are flies who've been fed on food containing these kinase inhibitors, and the kinase inhibitors appear to bring back the activity to far more normal levels. So as a way of testing drug candidates, of which the drug companies have many, by the way, this is a cheap and easy way of at least checking that these candidates don't kill living things and that they appear to bind to the right locations. We can also use machine learning, or what used to be called in the past statistics, to examine whether we can discriminate between different genotypes of flies carrying more complex or more unusual mutations that lead to Parkinson's disease. So by collecting multivariate statistics, essentially by probing these visual systems of the flies at different spatial frequencies and different temporal frequencies, we can generate many numbers that characterize the fly's visual system rather than a single one, which is its sensitivity. And if you feed those many numbers into multivariate classification algorithms, you can ask the classifiers if they can distinguish, if they can classify between different Parkinson's and wild-type fly genotypes. So here, for example, now is a grid of responses collected from normal flies and from flies carrying a Parkinson's mutation, showing the activity at combinations of spatial and temporal frequencies. And overall, again, you can see that the Parkinson's flies are exhibiting this significantly increased activity in their visual system early in their life. We can do this across a range of flies carrying mutations. So for these ones on the left are Parkinson's related mutations. These ones in pink here are mutations of the neural, of the fly neurons that don't necessarily model onto Parkinson's disease in humans. And these ones on the right are a collection of different wild type flies from different labs across the world. These should really all be the same response, but because of genetic drift, each lab now has a slightly different population of wild type flies. So our classifiers can accurately distinguish between pairs of genotypes, even genotypes that are rather closely related. So these almost very similar mutations in the DJ1 alpha and DJ1 beta genes. They can also distinguish, for example, between wild-type flies and flies carrying between different types of wild-type flies. So these are flies that once had a common ancestor back in the 1950s, but which have been carried by graduate students or postdocs to different labs and used to form different colonies, and which now are sufficiently different that they can be told apart by their responses to visual stimuli. And finally, we can distinguish between, for example, Parkinson's disease causing genes and diseases that affect the fly nervous system, but don't map onto Parkinson's. The clinical applications clearly are that you'd like to be able to diagnose Parkinson's disease from some sort of visual evoked response. And we think we can do that in these flies. So if you do a two-way classification between wild type animals and animals carrying any form of Parkinson's disease, we can perform this classification at about 86% accuracy, where baseline would be 50%. So we have some way of diagnosing a fly's genotype based solely on the multivariate responses that we can collect from its visual system when it looks at flickering lights on the screen. And finally, rather gratifyingly, if you map these multivariate data back down onto 2 dimensions using multidimensional scaling, they cluster in a satisfying way. So all the Parkinson's genes cluster over here in the manner that they affect the visual responses. These two nervous system but non-Parkinson's genes cluster over here. And all these slightly different wild type genotypes cluster over here in this little group. Finally, the story that we're coming to, I think, is that these mutations lead to relatively increased activity in early life, but that increased activity comes at a cost, which is increased excitotoxicity, increased sensitivity of the neurons to visual stimuli, and ultimately decreased activity in later life. So if you raise these flies in a disco ball, in a high strobing light environment, what you find is that initially you see these high responses, but towards the end of their lives, all these Parkinson's-like genes cause decreases in neuronal function. And more generally, I think the story that's coming out of this type of research is that Parkinson's disease may lead to increased neural activity early in life, and perhaps with some sort of advantages to the organism early in life. There's a hypothesis that it might lead to slightly increased intelligence or tolerance for white-collar work in some way. But that comes with a cost, and the cost of that is increased excitotoxicity later in life. So there's a trade-off in these genes between performance in younger organisms and performance in older organisms. And I think this is a story that's also being recapitulated somewhat in the Alzheimer's literature as well, that some of the genes that contribute, that are linked to dementia may also generate increased or better neural activity earlier in the organism's life. So I'm almost finished here. I'd just like to say one last thing. So we've talked about how we could take human genes and put them into flies. We've talked about how that could be used to develop some sort of pharmacological intervention and understand what's going on in these animals, but I haven't talked about closing the loop and bringing it back to humans. And the question here really we'd like to know is, do humans also exhibit these stereotypical changes in neuronal activity that we see in flies when they have Parkinson's disease? That's not an easy question to answer in Europe because the incidence of G2019S mutations, particularly where we are up here in this little weird island in the North Sea, is very low. But the incidence in North Africa, all across the bottom of the Mediterranean, is very high. So for example, in Tunisia, about 50% of the patients in a Parkinson's clinic will be carrying this mutation. So we were very fortunate to be able to go to Tunisia and make some measurements there. So here's Tunis, and in the Neurological Institute in Tunisia, we set up a whole, this is a nice physics thing, right? We set up a whole EEG recording system in that clinic. We had to design a whole sort of new technique for recording from people without electrode gel and some other things I could talk about later. But the long and short of it is that you can bring patients into the clinic and record from their heads as if you were recording from a fly with almost identical stimuli on the screen. And when you do this and you apply these multivariate decoding techniques to those patients, you're able to accurately distinguish between patients with Parkinson's, some forms of Parkinson's disease and controls. So for example, we can distinguish between Parkinson's disease and controls at about 66% accuracy, with 50% being chance. We can distinguish between idiopathic types of Parkinson's disease and controls even more accurately, and between idiopathic Parkinson's and these G2019S mutations still above chance. So to conclude, I think flies are the future. Flies are just great, and they're fast, they're cheap, they're easy, they're ethically unconstrained, and you can use them to model many forms of neurological disease, not just Parkinson's. They're rapid, inexpensive. SSVMPs in flies assay neural function just like they do in humans, and with some tricks in the frequency domain, you can even use them to dissect different levels of the fly nervous system. So it's kind of nice. Both fly and human SSVPs provide rich biomarkers for neurological disorders. So this multivariate classification procedure allows you to do some really quite nice diagnostics on both types of animal. And finally, this translates to other diseases. So we also have, I could have told you an almost identical story about autism. There's a paper we had with Dan Baker, who spoke yesterday, on doing this with autism rather than Parkinson's disease. And that's it. Thank you.
Thanks so much, Alex. That's absolutely fantastic work. I'm tempted to ask you if statistics really is the same as machine learning, but we've run out of time and it might crash the internet. You've got a couple of questions on the Q&A. I think instead of going to those, what I'd ask you to do is if you would have a look at those later and get back to that particular person. What I'd like to do now is go to the discussion panel. So invite all of the panel members to come to our virtual stage to unmute visually and through audio. And let's see, and then we can go from there. And this includes, I should say, not just the speakers of this session, but all panel members interested. Okay, so I wonder, maybe a starting point could be, Alex, where you talked about flies, and you suggested that flies are, fantastic, a really good model organism. And forgive my asking such a dumb question here, but can you walk us through how true you think that really is? How, how general are the results that you get from flies, both in normal behavior and under pathological conditions? How can we apply that to humans?
I get asked this a lot, and I think my answer has changed a little bit over the years. But I think if you care about things at the neuronal level that affect the way that neurons talk to other neurons, I think that flies are just great. I think that the neurons in flies operate rather similarly to the neurons in human beings, and that things like this gene that clearly affect them at a very low level possibly produce rather similar results across the two species. We've also looked at epilepsy, and that's another situation where hyperexcitability and moving into these sort of hyperexcitable states seems to be very similar in the two animals. We see almost identical EEG responses again in the flies as they are driven into epileptic seizures by, for example, banging them or strobing them with light that you'd see in humans as well. We're obviously not flies, and things that are more complicated, I wouldn't expect to see, for example, I don't know, grid cells or place cells or any of those more complex things that drive behavior in flies. So I think for diseases that we think are rather low level and affect all the neurons in the brain, I think they're nice little sort of compact behaving bioreactors that are a step up from cell culture. But I think for complex behavior, they're not going to cut it.
I wonder if I can bring Alessandra and Remy onto this, because is there a disconnect between that very complex level of a fly, a living fly, and a fly is obviously a reduced model organism compared to the human brain, compared to grid type modeling, for example?
I can give a shot at that.
Sure.
In very hand-waving generic terms, I think in the evolution of the nervous system, that for me is absolutely fascinating, is a main drive of science. That there are, of course, many different timescales. The nervous system has accumulated changes over different timescales, with a striking dominance of very slow timescales. So much of what we see in our nervous system of us humans is hundreds of millions of years old. But still, within the slow timescale of hundreds of millions of years, it makes a difference between patterns or phenomena that are preserved, say, for 500 millions and for 200 millions. There is a major difference. Mammals have differentiated some point between, I don't know, 250 million years ago, around that time. And there are things that we see only in mammals. There are other things that we see in mammals, reptiles, and birds, and not in the so-called anamniotes, among the vertebrates, those that did not come out of the water, let's say. And there are these other sets of phenomena observations, like fascinating things that we heard from Alex, that seem to have been conserved even, before the differentiation of vertebrates from insects and who knows what. So neuromodulators are among the things where you can find conservation of a very long time scale. It's amazing and fascinating. It would be really nice to be able to kind of study formally these very slow dynamics of what makes certain developments so resistant to change, and others instead more amenable to change. So I think no, it's a mix of things that change, things that stay constant, things that change abruptly. The hippocampus about which we spoke is apparently what we see is that all mammals have the same internal organization of the hippocampus, and no non-mammal has it. So the observations of modern species are compatible with the hippocampal internal organization having evolved all of a sudden. All of a sudden, maybe over tens of millions of years, but on a very distinct lineage. Before we lost, there are these kind of simplified mammals, the monotremes, that those that make eggs, and those branched out at a rather early stage, but still their hippocampus is like ours. So the evolution of the modern mammalian hippocampus is like a catastrophic event, or very beneficial catastrophe in the sense of evolutionary theory. Mm-hmm. And I think it's fascinating. But anyway.
Very good, thank you.
Can I add something on this point?
Of course, Rami, yes, please.
Yeah, no, I just wanted to say that even if actually, I agree with Alessandro, the organization can be quite different in mammals compared to other organisms. Some of the very basic conceptual, mechanism for computation can be also present in flies. For instance, the one I talk about continuous attractors. Mm-hmm. There were beautiful results a couple of years ago, 2017, by the Jaya Raman group in Janelia about the ring attractor in flies, for instance, which is beautiful. Of course, this is not hippocampal or grid cells, but of course, but this is interesting. And then I have another comment, and this is related to what Alessandro just said. I've heard about place cells also in pallium, in fish, in goldfish. So even if the organization is not the same as the mammal hippocampus, can you confirm, Alessandro, about that, or you think it's?
Yes. There is this amazing thing that, let me say this because this is too beautiful. Fish have a different developmental process, so that the region of the original embryo, the neural tube in the embryo, that in mammals and in other higher vertebrates becomes the hippocampus, which is medial, is like, I don't know if you can see me, is at the part of the cover of each hemisphere that faces the other hemisphere, is the medial dorsal part of the pallium. In fish, because of the way the embryo develops, goes to the lateral side, the lateral aspects of the brain. So the equivalent of the hippocampus is in a completely different place. It has a completely different organization. And yet there is this group in Spain, in Sevilla, that have done experiments in which they've lesioned the kind of quasi-hippocampus, the homologue of the hippocampus in fish, and they find deficits in spatial memory that are amazingly similar to the deficits you can find in a rat. They've done very nice things, like experiments in which you put fish in a pool and it has to remember how to escape from the pool, like the experiments that people do with rats and mice. So it's a mixture of things that are conserved and things that are changed, and it's fascinating.
Yeah. I was conscious of one thing that Fred said earlier. I think more about the limitations of experimentation, where the limitations were in optogenetics, for example. And I'm far from being an expert here, but I know there's a lot of work done on C. elegans, for example. But what I'm less clear on is how that sort of work can really be scaled up to the level of a mammalian brain, for example. I don't know, Alex, or even Fred, if you're still there, do you want to come in on that?
Oh, okay. So it's a tough one. Yeah. So two things. To the neural engineers like Dijsselroth and his cloud and the people that he trains. Currently, the approach is still there is no limit. We want to control every neuron in the mouse brain, and we'll just keep on improving the technique until that works. And the progress over the past 10 years has been phenomenal, unpredictable. So I don't know where to advocate that there is a limit. There must be a limit. So the second thing that I find noteworthy with respect to interfacing complex brains is, think of it in comparison to a sensory stream, like a sensory pathway that interfaces with the cortex. This is a tiny fraction of the inputs received by the cells. So even the direct recipient cells in the sensory cortical area, like the layer 4 stellate cells, they are a small fraction of the cells there, and they receive only 5% or so of their synapses from the input. So what that suggests to me is that in order to control a neural circuits, even a very complex one, in the sense that you have all the control that is needed for cognitive interfacing, that doesn't require complete control and command to every neuron. That can be with a very mild modulation. And so from that perspective, learning to interface with complex nervous system is very similar to learning how is actually information stream put into these recurrent circuits. So that relates back to the question of dynamical systems and chaos. Yeah? So something in evolution will have pushed the dynamics of neural circuits towards a phase that can actually be controlled. If they were wildly chaotic, it's not the useful kind of device. So because we are around and we have been weeded out over these 200, 400, 600 million years, that has tailored us to be controlled by very limited means. So hopefully that is what keeps us in business, or will bring us in business even more.
Alex, do you want to go?
So there's one more comment on this question of why the fly is basically another human. Yeah, so I like that a lot. And so it's super plausible for many neurodegenerative neurological diseases if they are basic cellular machinery. So, and that's actually what also our colleagues here in neurology, what the biomedical researchers are deeply convinced of. So if something really goes wrong at the cellular level, of course cognition will be affected and memory will be affected and so on. But so many of the colleagues in biomedicine are actually convinced that you don't have to fully understand memory recall to understand dementia. It's the disease is a disease at the cellular level. If they are true, there are so many things that we inherit, we have in common with sponges, all the synaptic proteins of them, half or more are preserved from single-celled organisms and from the earliest animals. And then the best and the ethically adequate animal model to study are those very simple ones. And flies are amazing in their cognitive abilities. So you could even go to a sponge, but it's hard to do a cognitive task with that. So I think there's every reason to believe that Alex's philosophy is super on target.
Mm-hmm. Alex, do you want to come in on that?
Yeah, just to exactly mirror that, it depends on the question you're asking. It may strangely be a fact that it's conceptually much easier to fix some of these terrifying brain diseases, much easier than it is to understand how a normal brain does something very simple like navigate a maze. They may be orders of magnitude different in their complexity. In one case, you're really just understanding what a very simple cellular deficit, what that's doing to the network, and just fix it. But I think, as the talks this afternoon have shown, getting any sort of handle on the dynamics or the population dynamics of real cognitive processing is just astonishingly hard. And I wouldn't look to flies for that, but I would look, as you said, to simple animals for simple models of genetic disease.
Okay. So actually going back to one of your slides, Alex, you you alluded to sort of molecular length scale effects, and I just wonder, I wanted to tie in with something that you said, Fred, about the sort of limitations, experimental limitations, maybe through instrumentation, and maybe could you walk us through that a little bit more? Could you explain where do you think we're at in terms of the current limitations, and what do you think would be transformative if you know, a group of experimentalists could develop a bit of optical microscopy? What would it be that would really transform insight here? So maybe back to Fred on that.
Yeah, I'm really out of my depth probably for a real answer to your question.
Yeah.
But so that, and it's going to be not one thing. So for the question that Alex asked, much more, and that Viola showed some theory about. Better a next generation of in vitro models. It can be really so simple that you may not count it as instrumentation, which means to get the scope and the stimulation apparatus inside of the incubator, so that you don't have an experiment in that sense, but still have complete control and can handle the data rate. So that can be for drug screening and so breakthrough kind of progress. Although it's not interfacing with the brain or interfacing in brain-like fashion.
Yeah.
In order to interface with a living neural network in a way that can be compared to a neural projection, an advantage is that many opsins are like excitatory neurotransmitter receptors. An AMPA receptor is basically 0 millivolts reversal potential channel, and that is channel rhodopsin as well. So you can think of channel rhodopsin as a photon selective neurotransmitter receptor if you want, and you can emulate a synaptic AMPA current by the right kind of light stimulus. The challenge is to address that to a chosen set of cells. So one has to sculpt the light such that it addresses an individual neuron, which means it has to be done through the soma, because the dendrites are intermingled. There in this neuropill you cannot individually address a neuron. That can only be done if you stimulate through the soma. And so that technical challenge has two aspects of it. Getting the light down there to a 5-10 micron spot that you target. You have to first map where the neuron is, and then in your situation get the light there and get the opsins concentrated on the soma. So the second part that has been partially solved by geneticists and cell biologists. There are tags that bring channels of that you bring genetically into the cell selectively to particular compartments, to spines, to the soma, and so on. So combining a technique like holography, 3D computed holography, with targeting the opsins to the soma can give you single cell resolution individual access. But if you think of 10 cells and you want to pre-compute the holograms for all the 10 factorial different stimulus patterns that you want to project, that is going to be hard. And the larger, so that you will not exhaust this repertoire of activity patterns that you want to send into the stimulus. And you have to be smart and elegant with the computing that goes on before you can do that. So that's a stimulation design problem that is that mirrors these maximum entropy modeling of neural activity patterns. So there's so many things we cannot exhaust if we sample all the stimuli that we want to pre-compute. So that yeah, there must be a rational choice of the ways you want to really feed into 10 neuron, let alone a 10,000 neuron population.
Yeah, yeah. So I was just wondering, you know, how valuable could it be if you zoom in even further? So you mentioned the level of the single cell, but then, you know, you zoom into the level of the synapse. But then I guess what you'd really want to have then is information from other synapses synchronized, I suppose. Is that correct?
There are interesting and computational and biophysics questions at all these levels. So for the single synapse or for a small group of synapses on the dendrites, that makes sense. And so imaging that has been there's been a lot of progress. So tuning imaging of individual synapses, also stimulating single synapses. Presynapsis has been done. The question I can think of, they actually, then I would think of like one spine and its plasticity roles in vivo or in the intact network, or multiplicative interaction between a couple of synaptic inputs into a short segment of a dendrite. The postsynaptic side, I think, is where a little bit more progress needs to happen.
Okay.
And the reason I say that is, so for one question postsynaptically for neurons like in the cortex with spine synapses was whether the spine is just a chemical compartment. You want your local calcium signal in the spine head, and that shouldn't bother to the rest of the dendrite, or whether it's also an electrical compartment. Depends on, so the critical unknown was the spine head's resistance. If basically electrically the spine head is short-circuited with the dendrite, it just follows the dendritic voltage, but can it break free? And for like 20 years, so Francis Crick a long time ago hypothesized that the spine neck might be so thin that the spine is an electrical compartment, and then different computations are feasible. But that came out of so there was no evidence supporting it. That just came back. So Rafael Juste and co-workers managed to get voltage imagings to such a spatial resolution that they can start to image the voltage of the spine head, the spine neck, and the dendrite. And in one paper in Neuron two years ago, they reported first strong evidence that the spine head is a separate computational device. It has interesting physics in it because you can recapitulate it with models only when you take not cable models, but Poisson-Planck equations, so that there are that you have really a charge distribution, non-charge equilibrium, so there's they hypothesize, they actually give computational evidence that there's a kind of skin effect in the head in the neck of the dendrite. That's like 50 nanometers, 100 nanometers compartment, and if you have charge surplus there, the charges are pushed to the membrane. So that gives okay, I'm getting carried away because I find it fascinating. But at this very small scale of nanoscopic compartments, there's a lot of cool physics that's becoming feasible. And with optical interfacing, one can do real physics level experiments with these subcellular computations. So I think that's a great point that you mentioned.
Brilliant, thank you. I think we should probably draw that panel discussion to a close. Thank you so much. It's been absolutely fascinating and enlightening. And I'll hand over now back to Tom McLeish. Thanks, Tom.
Well, Mark, thank you very much, and a big round of applause virtually for today's panelists. That was absolutely wonderful. I'm reeling, and wasn't there Nagel, the philosopher, didn't he? Said we'd never know what it's really like to be a bat, But Alex, thank you for helping us realize that we might be able to understand what it is to be a fly, or at least the other way around, particularly as we get older. So let's hope the best there. So many thanks. I just have a few things to say to everyone. Nobody go away, because there's more fun about to happen. If you've always wished on these Zoom meetings that you've attended since March, if only I could do that nice thing where after the conference we all file out of lecture theatre and go to a nice drinks reception. Well, you can, virtually of course, but it'll be just as fun. Follow Karis's link to the Wonder thing in 2 minutes time. I have something to ask you about first. Final message. Come and find out more, either on Twitter or Physics of Life. There's loads more workshops, including following on perfectly from this very session we've just heard. Rounds, Trees and Physics of Life workshop on the neurodegenerative diseases of the brain of both human and fly, no doubt, and others. Please sign up, come along. There's the website, find out more. Now, as I was just saying, come to the meet and greet in 2 minutes time. This is a slide we mostly showed yesterday. Karis will have sent you the link. Safari, Firefox or Chrome are best. Type the link, enter the password, in you go. Hot tip we didn't say yesterday: when you get teleported into this room with tables, a big room, the first scene you get is only your immediate location, and you might find yourself a little bit alone. So the first thing to do is to work out how your machine to zoom out. On a Mac, it's 2 fingers on the board and push up, and you zoom out. You can see everyone, see all the rooms. What we'll have there when you first get there, as I said yesterday, we're going to suggest for half an hour or so, we do a little bit of gentle fun work. So when you've got your glass of beer, wine, fizz, pop, Coca-Cola, nice cup of tea, you will find 3 sort of areas, 3 discussion areas. When you go into those areas, what we'd like you to do is to take what we've been discussing the last couple of days and help the conveners of those tables to answer little 3 questions. So what are the new interdisciplinary questions that are really ripe, that you've got excited about over the last couple of days for life science, physical science, medical science collaboration? So what are the questions? Just the questions. They could be and sub-questions. What methodologies might be combined? We've had lots of chat about that this afternoon. And who might we be able to help, particularly people who we've not seen here, but we know about, who we might contact. And here are the 3 areas of questions. So I'm going to be in 1, Mark's going to be in the 2nd, and Steve Smile will be the 3rd. Anthony Morland will be roving around the room, helping people and helping us all out as well. So I want to follow these questions related to scale, renormalization, structure, dynamics, information processing. How can we probe that experimentally, theoretically? Does this idea of scale invariance help us understand how fMRI can tell us anything? And what might it tell us in the future? Second, there's a whole question related to structural wiring and implications for medicine, and Mark will be looking after that little table. Mental health implications as well. Overlap there, obviously, between all these 3, and questions on nesting and hierarchy with Steve Smyre. Again, there's a huge amount of overlap here, but these are ones to start with. If you've got other questions, it occurs to me, for example, that we haven't, apart from this last session and this amazing panel, have really addressed questions of evolution, and it may be that there's an opportunity to feed some of those questions in as well. I can see Karis has just emailed around that link as well. So that's all we need to know. I suggest we might beaver away at these questions with these 3 conveners till about 5 o'clock, and then think of socializing about on other topics. So that's all from me. It remains for me, on behalf of the other panelists, Karis, to say thank you very much indeed, everyone who's contributed, the Q&A, people who've turned up, speakers, the panels. It's been absolutely riveting and very, very stimulating. Thank you so much. Thank you once more to both Karis and Steve Parker, who've both been in the background, without whom the whole thing would have fallen apart within 5 minutes yesterday. Thanks very much for holding us together. And with that, we'll all see you in the Wonder Room, unless I've forgotten any vital thing. And I should have, yeah, brilliant work. No? Good. Right, we're off. See you in the Wonder Room. Bye-bye from Physics of Life.